Okay, good morning everybody. This is Schmidt Ocean Institute, Research Vessel Falkor, ROV SuBastian. We are now live on YouTube and Facebook. Thank you for joining us this morning. This is ROV Dive 366, and we are on the north side of Lihou Reef in the Coral Marine Park in the Coral Sea off the east coast of Australia. And you're currently looking at live images of a Nautilus swimming. So thank you for joining us this morning. We'll be joined later by Dr. Robin Beeman, our chief scientist, who is at his home in Cairns. And we are going to head back down to the bottom and get started on our transect. For those of you watching at home today who are used to seeing our typical ROV overlay, it's not working, so you will not get a depth. And it is currently 497 meters, and we'll be starting around 520. Okay, good morning everybody again. Sorry about that. A few little technical difficulties this morning. As you can see, we don't have depth or altitude overlay, so we're working on trying to get that fixed for you. But we may or may not have it done today. So in the meantime, it is 548 meters for depth, and we are just taking a look at some of these geological features. I'm going to change your screen for you, and as you can see on the lower right hand corner, this is a Google Earth display. This is the Queensland Plateau. So this whole area is where we've been working, and you can see the lighter colors, pink and green and blue, are areas that we've been mapping with the multi-beam. So if I zoom in over here to the far east corner, southeast corner of the Queensland Plateau, we can see this is Lihou Reef. And we've done just one pass on the south side. We've had quite a bit of weather over the last 3 days. And so yesterday we were able to duck around the corner and get one single pass on the south side. But for the last couple of days, we've been working on this northern side of the reef. And if we zoom in where the ship position is, this is our dive location for this morning. So we're working our way up towards this key, and we can see sand out the window. I went by here the other day just to check the setup, and so this is exposed above the water. And this Nautilus just wants to come say hi. And so this is the 3D plan here for the day. Ah, there we go. There is your overlay. So I think we have, Daniela, are you there? You should just have to unmute yourself.
Yeah, I'm here. Good morning, Debs.
Good morning. Thank you for joining us.
Can you hear me?
You should just have to unmute yourself. Yeah, but you're going to have to pause
Yep,
your
done.
you're going to have to pause your YouTube. So go ahead and close YouTube and mute that. There you go. You should be able to see what we're seeing here on the Zoom screen. And just remember, it might be just a couple of seconds delayed from what the ship is seeing, but it's up to about...
Yeah, I'm here. Good morning, Debs.
Good morning. Thank you for joining us.
Can you hear me?
You should just have to unmute yourself. Yeah, but you're going to have to pause
Yep,
your
done.
you're going to have to pause your YouTube. Daniela, I think you can probably hear me now. Sorry, my Zoom was muted and yours as well. But yeah, make sure that everything on your computer for sound is off for any of the video.
Okay, I've done that now. Can you hear me?
Yeah, I can. Sorry, and I didn't realize I was muted. So, good morning, everybody. Welcome to our technical challenges with chief scientists and co-PIs shoreside. So thank you for joining us. Why don't you tell everybody a little bit about who you are and what your work is, and then we can talk a little bit about what we're seeing.
Sure. Yeah, thanks for inviting me on, Debs. My name's Daniela Ciccarelli. I'm a consultant, and I do mostly some freelance research work with universities and government agencies and research institutions. And I've done a lot of work on the shallow coral reefs in the Coral Sea, since about 2007, when I was first contracted by Parks Australia, as it's now called. Back then it was the Department of Environment and Heritage. And just to survey the shallow reefs, because I mean, what we're seeing here is completely new, but even in the shallows, the coral sea is relatively unexplored. And even though coral reefs generally, behave a certain way, there are unique aspects to the reefs in the coral sea that we don't really understand yet. And for most of them, we actually don't really even have much of an inventory of what lives there. So for the last 3 years, Parks Australia has contracted James Cook University and the ARC Centre of Excellence for Coral Reef Studies. And I've been very fortunate to be on the team to survey the reefs, the shallow reefs of the coral sea. So we've done these long expeditions going from the very southernmost reefs all the way up to the top to Osprey Reef, actually. And one expedition, which I unfortunately missed, went all the way up to Ashmore and Boot Reefs. So I've just been involved in exploring the shallows, and now I'm feeling very privileged to get to watch the exploration of the depths.
Yeah, it's definitely interesting to see the two contrasts, right? From what you dive on and see a lot more color, I think, up there. From what we've seen a little bit, we've been able to come up at reef height, and everything looks very brown and green down here, with the exception of some amazingly colorful coral.
Yeah, I mean, what strikes me down here is that you have these seascapes that are very uniform in color. And yes, as you say, brown and green, but often the life that pops out, even though it's fairly sparse, is often quite colorful. It's often red, right? Because that's a useful color at these depths for animals to be. So you sort of just get these little bright sparks, and so I find myself glued to the screen, waiting to see what pops out next.
Yeah, us as well. I mean, when we sat down this morning, first thing we had 2 Nautilus join us. So we've had some amazing critters sneak up on us, unbeknownst to us. There's an aft camera occasionally that we'll see a shark or something in. And so, yeah, it's really really fun to see what's down here. And I also find the geology of all of this really fun, from the mapping to being able to kind of see the boulders themselves as we set down on the reef.
Yeah, well, and I mean, there's always, even down here, there seems to be a really clear link between the geology and the biology, right? So as soon as you have some structure, there's things for animals to hide in, there's things for sessile animals to attach to, and that means there's prey for bigger animals. And so more complex geology tends to attract more life. And that's, I think, true of these depths more than anywhere, because you can get these, from what I've seen, quite vast expanses of very little structure, and then structure becomes extra important.
Yeah, definitely. We always, when we're dive planning, with the science team and everything, everybody's always looking for, as most of you at home have heard Rob say, deep and steep. And but also really what we're looking for in that steep bit is some rock face. So we, over the course of all the expeditions Falkor's done, we've definitely seen a trend to, the amount of current and the amount of geological structures for coral and other life to live on. But I'm always amazed too at things that just live out in the sand, that it can just survive all by itself, what seems to be a wide open space with nothing there.
Yeah, you're right. And also, it seems to be so unprotected out there on the sand. But I guess that's testament to how sparse the predators are too, right? So I guess if things are able to survive in the open like that, it means that they, the risk of getting eaten, or they might have some other mechanism like sucking down into the sand or burying, or, they have some other technique of protecting themselves. Robin's just sent in a little text saying these are sponges, rock sponges. And sort of looking at them and going, wow, you know, like in the shallows on the really exposed sides of the coral sea reefs and Lihou Reef in particular, you actually get where it gets really hammered by waves all the time, and it's actually very flat, and it's like a moonscape. The reef's like this rolling plain of moonscape pavement. And the sponges that, like the dominant benthic form there, are these sponges that look much like these ones. So it's really interesting. Little grey sponges. I'm not a sponge expert, but I'm sure they're very common and commonly known. But they look like these ones here. So that's pretty amazing. The parallel of what's growing deep and shallow.
Yeah, it's definitely the diversity is really interesting. And I think every dive has been very different. Well, not very different, but has definitely been different for us, depending on the reef and how deep we start or how much geology we see. And I'm actually surprised. I'm just going to bring up a different image on the multi-beam Fledermaus overlay, because you can see here now as I zoom in where the ROV is, and we're just looking at this little bit of a channel, and so we're seeing the wall face of that. And so it's interesting that it is quite as rocky as it is. So I'm sure Robin and Jody will have a lovely conversation about that later. And it's a lot of fun to sit and chat with them about why and what creates. So as you can see, there's this huge slump area, and we're going over this debris boulder field and working our way up to this sort of corner where this whole side is sort of fallen off the side of this K. And so we're hoping eventually, once we get up towards the end of the dive and up on the face, to see actual sort of the striations of the reef. At least that's Jody's hope. And then be able to kind of, just see the different geological structures as we come up. But
so which of the Ks are you? You're on the northern side of it, right? So
yeah. So if I
know
zoom,
the name of it?
when I looked on the chart, it wasn't named, but the two, I believe this was, I think the two or three to the right, the big one in the corner is named, and then the two to the west of that, but these two didn't seem to have a name. It just listed it as K. So I'm not sure, maybe if there's a different chart or naming convention or place that gets saved, but I looked at the nautical chart and it wasn't on there.
I'm going to have a quick look at some of my maps and see if I can find a name, because yeah, you're right, on different maps and nautical charts, some of them have names and some of them don't. We certainly, when we were looking for sites to dive on, we were using those names to name our sites. So I'm really familiar with the named reefs and Ks.
Oh, nice.
And not so much with the let me see.
And just to give you, I'll put it back up. I think we're going to zoom in on some coral here and a little blue fish. But let's see here. I'll move this over. We were able to do a little mapping yesterday on the south side, and so if you want, I can give you a sneak peek at was Kate K was the one you were talking about in the call the other morning?
Ann K.
Can K, okay. And that's here.
Ann, like a name, like a woman's name.
Sorry, Ann, yeah, Ann. Ann K, so that's this bit of mapping that we were able to do yesterday.
Yeah, we were talking about how, there's a little bluefish. Were you talking about this bluefish or
yeah, this bluefish?
Well?
Nope, this bluefish.
Is it evading? Is it swimming backwards out of the light? It's backing out. Nothing to see here. I'm out.
Yep. You can look at the coral. Just don't get distracted by the fish. Well, there's like a see-through one in the right lower corner. Huh.
Oh, yeah. We need Kai here to identify them.
Yeah, and then Kai will say he doesn't do the deep fish, so
Well, neither do I. I'm usually in the shallow coral reef surveys. I'm the fish person. So, but yes, also finding myself kind of going, what? It looks like it looks like it could be an epigonid, and I know that there's actually deep sea cardinal fish called epigonids, which I've learned since I'm on board this project, and that actually did look a little bit like one that that Kai named on a previous slide. But you know, in the shallows, those fish are nocturnal, so out here they might be, or down here, I should say, in the dark, they probably be well in their element. But I just wanted to go back to this NK spot while we're waiting for something else to pop up. We were talking about this the other day when you're looking for places to potentially dive, weather permitting. And oh, there's another one. Yeah, it looks like an epigonid, one of those that Kai identified the other day. I could be very wrong. It'll probably come up on the YouTube or Facebook chat if I'm wrong. There'll be experts there naming things.
Yeah, they'll jump in our fish army.
Great. Look forward to seeing what they have to say. So what we're talking about with Ann Kay is that on our shallow surveys, that seemed to be a spot that had a really big aggregation of diversity. The corals seemed healthy and dense. There were a lot of fish. There were things spawning. There were aggregations forming of fish spawning. In fact, while we were there in early March this year, late February, early March, there were big spawning aggregations of surgeonfish, of acanthurids. In fact, there was this ball of about 1,500 unicorn fish. These are types of surgeonfish, and they're planktivorous. And there was this ball of 1,500 unicorn fish being herded by sharks, by about 20 reef sharks, right in front of our eye, on this very drop-off that you're showing there in the map. And so there must be something going on there.
Well, it's definitely
in terms of currents or eddies.
Interesting geologically. I was just saying it's definitely interesting geologically because, one, you can see that there's definitely some sediment buildup, but as it's sort of around the corner, that reef face looks to be quite dropped off. It's quite deep and steep here. And then you can see as the sediment, it's actually being pulled around that corner. So the current there must be pretty strong as that flow is coming down and then turning around towards the north, actually. It's not full north. This is north up, so going to the east. So it's sort of coming down, but then also flowing around the corner to the east. So it's definitely interesting.
Need to be carrying that sediment away and like scouring out the rock face there.
This would definitely. So hopefully we will. We have to do another check on the weather, but hopefully this might be our dive for tomorrow, somewhere in this area.
I'll be very curious to see what's there. That's cool.
Yeah. So for those of you at home, we are actually still, that are just joining, we are working on Lihou Reef. If I zoom out here a bit on Google Earth, give you a little bit of an idea of where we are. There we are, working in the Coral Sea on the Queensland Plateau. And so we're way off to this eastern side. Working up on this northern corner.
It's like a graveyard here, Jim. Nautilus graveyard. Yeah, I think so, yeah.
Oh yeah, they're just noticing that our ROV team and our other lead tech Jimbo, they're noticing these are dead Nautilus shells, so it's quite interesting. This is the most we've seen all in one spot, so it's quite interesting.
Would you usually see them on the sediment like this falling off the side of the steep?
We have seen one or two.
Side?
We've seen one or two like this on the bottom, and actually we saw some. We actually saw some. We saw one live one sitting on the sediment. So they seem to also at times rest on the ground, and then sort of go up and swim. So that was the first time. I think actually Rob had said that day that it was sort of the first time we'd seen it sitting in the sediment like that. But yeah, there's tons of them here, and so this is the most we've seen in one area really kind of be congregated and to be old shells.
Yeah, that's interesting. Do we know what's the main source of mortality for Nautilus? Do they get eaten, or
they do get eaten by? Sorry, they do get eaten by octopus and squid and stuff, but I don't know in terms of what the commonality or the rate of that is. Maybe we have some Nautilus experts on. I think Andy's been on YouTube on and off, so it'd be interesting to know what they think of this area with all of these sort of dead shells.
Do you think they could also be washed in by the current, by the surrounding area? I found Lihou's interesting because there's not really, like, how most reefs you'd expect there to be a sheltered side and an exposed side, and they have characteristics that are more or less typical in terms of shallow reef ecology. But on Lihou, there's not really much of that going on because the southern side of the southern edge is exposed to the trade winds and storms that come through from the southeast. The northern side of the southern edge of the reefs has this long lagoon, this large lagoon, which is, it's large enough for there to be quite a bit of fetch. So when the wind's coming from the north or the northeast or the northwest, there's enough wave action there so that northern edge of the southern reefs is also quite impacted. So that sort of gets heavily scoured out to some degree as well and is quite complex. And then on the northern edge of the whole reef, the big reef, again, you have the fetch from the lagoon creating southeasterly waves that batter the southern sides of those reefs and cays. And then on the northern side, that's exposed to the monsoon northerlies. And so you really don't have a typical sheltered side of Lihou Reef.
Yeah, that's interesting. It's really interesting. I'm not sure that I picked that up quite specifically like that going around all these reefs, but it does seem to be, even from the LiDAR data that you see here, you're right, there's quite a lot more erosion and it's a little bit deeper. The purple color you see in the window is 50 meters. So it's quite a bit of a drop from the surface straight down right next to the reef. So you do see quite a bit of that type of, like you were talking about, longer lagoon erosion and things like that. And it's really interesting to know. And I find it interesting because this is up in the north, so yeah, there would or wouldn't be some protection there. And 1 second, Danielle, I just have to jump off for a second. I'll be right back.
Mm-hmm.
Sorry about that. Yeah, just a couple of things happening in the control room all at once. For those of you at home, this area is pretty flat, and as you can see, very sandy and slight bit of sand waves, probably from the current and stuff. So it should be probably pretty sparse here. Our video logging system just went down, so we're working on rebooting that as we speak. So we're gonna hopefully not find anything too interesting in the meantime as it starts its boot cycle back up.
I just had a look at the map of our shallow sites, and just to the west of here is Lorna Cay, which we've surveyed quite a few times, and AIMS, the Australian Institute of Marine Science, surveyed in, I think it was first surveyed in 2003. And then before that, Tony Ailing surveyed quite a few of these reefs in 1984.
Oh, wow.
So we have some information going back that far. But yeah, so these, we were just talking about how there's actually not much shelter. Usually the northwestern side of reefs here in this part of the world is considered the sheltered side, but these reefs are really exposed to the monsoonal northerlies. And so even in the shallows, this area is very sculpted, and big blocks of limestone structures sort of marching their way down the slope when you're on the edge of the shallow reef, diving at sort of normal scuba depths, and looking at these huge blocks of reef, and they're like big bommies, but like supersized bommies, like buildings, and there's a relatively gentle slope at that point, and there's just all these blocks marching their way down into the depths.
Interesting.
So this is right at the bottom, is it? This plane?
Yeah, it's we're sort of working on this. I think, let's see, check out. Yeah, so we've just come up out of that little bit of a trench where we were looking at the wall face here, and we're starting to work across this probably greater sediment before we get to the next rocky debris area.
Yeah, so it's interesting how life has to basically cluster around those harder structures where things can attach more easily and hide under and in, and I wonder how much movement there is across these sandy areas between those little patches of habitat.
Yeah, it's definitely something interesting. We see a lot of time in the sand. We see like sea stars and sea cucumbers and things that are very sediment driven, and they do seem to move quite a bit. You can see the tracks usually in the sand.
Yeah, look at that. Yeah.
There's some brittle stars that look like they're crossing the desert. And it looks like an anemone on the side of that rock. Great, thanks, Jimbo. Just got our video recording back up and running. We have a number of redundant systems, so we haven't actually lost video. We're right now redundantly logging with 2 different systems, so we get lots of video recorded.
Mm-hmm.
Here we go.
And then, so what happens? I think it'd be interesting to hear about what happens to all this footage afterwards. Like, who goes through it, and what happens? Are things cataloged and identified and quantified, or how does it work?
Yeah, so that's so it's a little unusual for this expedition because I think this was, the plan was maybe to do 4 dives, and the primary focus was mapping, and the whole expedition was put together a little bit last minute because of the ship having to reposition due to COVID-19 and having a gap and not having scientists come out. So typically the video data, and for every expedition where the ROVs use, the data and the video from the vehicle go with a science team. So it is typically something that's been agreed upon or worked out or figured out between that team of scientists on who's going to work on what parts and who wants which video, and where all of that's going to be published and archived to. So it's usually a lot of times very institution dependent. So at SOI, we do keep all of the data. We use a Google Transfer appliance, and everything goes up to the cloud for us. And then as people request video, we have somebody that works on that. So, but we don't have a full repository of all of the video data or images where one central location is that all of that ends up being.
Mm-hmm.
And we also do also use this stream to YouTube as sort of an archival method as well. So, you know, it's available to the public after every dive for all of time.
Great. Yeah, I mean, it's certainly a treasure trove of information that you're collecting here, and you know, to be able to, I just love that we can link it nowadays to the more shallow habitats as well. Like we have this, you know, complete track from the bottom to the top, because the top's been done, you know, by divers, and we have footage and photos of that, and now we can follow it all the way down to the bottom, and hopefully what we can do is start to identify some patterns of you know, what links there are between the shallow and the deep habitats, if any. I mean, it could be that there's a real kind of barrier because there's so many adaptations required to live in the deep that whatever's there is not really relevant to shallow life.
Yeah, it definitely, from watching this footage over, you know, even just this expedition, but also over the years, it does seem to be that there are these depth boundaries where you see both a change in biodiversity and also what that diversity is. You know, as you can see, like even the Nautilus, we see them, you know, in that 500-meter range, but we don't see them again once we go up above, I think, 350, 400 meters, and we haven't yet seen them, you know, really super deep. So they definitely seem to have a very narrow range of where they're happy at.
Yeah. So yeah, so it's interesting to think about the vertical connectivity that's going on here, especially since you have, you know, it's not out in the pelagic ocean where there's no structure. This is a definite structure that things can, you know, sort of orient themselves to. Is this another anemone?
Yeah, it looks like it. We've seen some really interesting, it looks like it's just in, maybe it's, we've seen some over the last few dives where these anemones seem to be sitting in somewhat of a little moat around them. So I don't know if there's something that is living with them and creating this sort of channel, or just the natural flow of how the water moves around it. This one looks like it doesn't have quite as much of a deep moat trench around it, but yeah, definitely something interesting we saw a couple of days ago on Dive 365.
Interesting that it's not reacting, because we do get similar anemones up in shallow reef areas where there's sand between the reefs, and you see similar anemones, but the minute you approach, they scoot down into the sand, they sort of disappear into the sand. So this one and the one we saw before, they don't seem to react.
Yeah, and I think it's because we approached them from the beginning, and we approach them from sort of far away, and we're still 1.5 meters or so off the bottom, and then we're zooming in with the camera. So they're not really feeling us until probably we swim over top of them. So as we see them, they're in front of us, and then we zoom the camera even more in front of us, and so we get to see them before they have a chance to react. If we were to turn the camera around or the vehicle around, you'd probably find it was potentially closed after we flew over the top of it. So we're going to set the vehicle down for a moment and take a look at some of these. I think there's a fish and a hermit crab and an anemone.
There seems to be a lot of life buzzing around the light, maybe. Are they copepods or other kinds of plankton?
I
Or is that on the inside?
Yeah, there's quite a bit. If you look back, if I change the, after we zoom in on this really
there's the
cool
fish.
iridescent fish.
Are you getting any IDs on YouTube or Facebook?
I haven't seen the usual crowd yet this morning, so, oop, and that fish
there it
just sort of
goes.
hid itself in the sand.
So there's a hermit crab. And do you see those kinds of shells down deep, or would it have depended on that shell falling down from shallower?
I think it's been a combination of both. I think we've definitely seen some shells, but usually, oh yeah, that's what I was going to do. Sorry. Chris just reminded me if I show you the sit cam. So if you look at this, I'm going to change it, situational cam. This is what's in the water column right now. So this is a huge amount of life going on in this water column right now. Unlike, I think, anything we've seen on other dives.
Oh, really? So this is unusual.
Yeah, it's not usually this dense. You know, we'll see some marine snow and we'll see a little bit of here and there, but this is almost whiteout conditions, as you would say.
And is it mostly alive, or is it marine snow as well?
I don't know if I can tell. We haven't really zoomed in, but there's definitely quite a bit of alive movement. You can see some larger stuff swimming.
Oh, yeah.
Yeah, Ben, can you look up with the science camera for a minute, and I'll switch the back. I know, but you can see some. Yeah, you can definitely see
that's quite
some
a lot.
stuff.
Yeah, that's quite a lot of sort of nutrient potential coming down. You know, marine snow, I guess, for viewers that might not know, is sort of organic material that's not usually alive that falls down into the depths from shallower waters. So, you know, anything from dead things to fish scales to just bits of organic matter. And in a lot of places, deep sea life depends on marine snow for food. But this is far beyond that. There's so much life in there.
Yeah, it's really fascinating. I think that's how, you know, a lot of these organisms can survive out here in this, what would be very deserty for us, but I think they do get quite a bit of deposit from both off the reef and the current flowing around this area.
So I've missed quite a few dives, or maybe a couple of dives, because I was out at sea myself with the Australian Institute of Marine Science in the last 2 weeks, and so I couldn't link in. So I'm just curious to hear from you, Debs, which dives have been the most exciting or different or interesting for you having seen them all.
Yeah, I'm not sure I have a favorite yet. I think for me it tends to go from, you know, more I like octopus, so I'm always going to remember the dives with octopus, and I think the dives that do this sort of start deep and we are able to get all the way up to the shallow bit are the most memorable in a lot of ways, where you really get to see that full range. But I also really do like the deep. I think, you know, even though it seems like it's a lot less stuff, it just has some of the most unique animals, and they're, you know, whatever they've chosen to be their adaptation to live in this environment, are just amazing. And so, you know, we see for us new things, but things we've seen on other expeditions in other places in part of the world. And so, you know, and they do kind of, I have to say, maybe this sounds bad, but they do tend to run together. So I don't know if I can pick one out over another, because after 5 weeks of 5 weeks of doing this,
get it.
yeah, after 5 weeks of this, and 5 months, I think it's definitely Even the expeditions tend to blend together, although they do tend to stand out in their own way. When I started in January, we were in Bremer Canyon, and now we've come almost all the way around Australia. So it's been fun to see
Fantastic.
the different areas.
Yeah, it certainly happens when you're at sea for a long time and you're doing you're doing a systematic sort of thing again and again, and those tend to all blend into one. But there's, usually moments, like you say, like depending on what you like, the octopus that maybe stand out, and the odd moment. But what strikes me too here in these deeper areas is that the animals seem to be so solitary, like in the shallower areas, life sort of depends on each other. Things depend on each other so closely, and things are kind of living on top of each other, and life is competing for space and light, and down here it's pretty much the opposite. Things are solitary and are probably need to be much more self-sufficient in terms of just their whole life history.
Yeah, it's interesting. I was editing the video from the last dive 365 just last night, and I sometimes don't see the end of the dives because I'm off in the middle of the day, and so I was mentioning to Jimbo, our lead tech, and saying, can you point out to me when the fish were there, because it's so diverse right now. I can't even tell where a fish would be in between all of that coral and sponge and the colorfulness and all of that. And so, it's definitely a big contrast between the individual organisms down here in the deep and how solitary they are versus just the plethora of life up on the reef.
Speaking of solitary, there's two here.
Yeah, these are interesting little, very shiny fish.
They are.
Chris says black dot shiny fish. That's about the level of our fish identification in the control room right now. Yeah, I haven't seen Kion today. I'm not sure what he's up to today, but he hasn't.
If you translate that into Latin, you probably get the name of the fish.
Yeah, that's true. I know when our fish army starts reading off all of these species and names and family names, I don't have any idea what they're talking about.
But actually, the scientific names of things are often a description of it in Latin. So, yeah. In fact, you'll often find something being the one-spot butterfly fish, which you might, if you were doing exactly what just happened now, like the black dot shiny fish, yes. You find that actually these are the names of fish. It's just they sound much fancier in Latin. Yes. So, whoever names species gets to choose what to name them, but it also does follow a convention so that they're either named after a place or a person, or they're named in a descriptive way. But it's usually pretty simple, and often, your gut feeling of what it could be called is often correct.
Yeah, this little shell, we've seen two of them. This is another anemone, I believe, but then on the lower right-hand corner, there's a kind of like a hermit shell, but it's spiky, and we saw one just a little while ago, too. They're quite interesting. I haven't seen the spiky ones before, or at least noticed them. Yeah, Jimbo was just commenting about every reef we see something different, and I think these spiky shells are different here than we've seen on the other ones. But most of them are Nautilus, which is
crazy. Has it got a crab in it? It does, doesn't it?
Know.
So it's not the original. Oh no!
It might be a snail or something?
So that's actually then the original mollusk, because hermit crabs pilfer dead shells. So usually if you see a shell on a hermit crab, it's not really an indication of that's where you would find the live snail. Sometimes it's fallen off the edge of the reef, or it's especially down in the depths. It could have come from much shallower, but or been transported by currents. Or hermit crabs also fight each other for the next bigger shell. So it's not always a good indication of it's a good indication of the presence of that mollusk somewhere in the general vicinity. But usually, if you see the live snail, it's a much better immediate indication of a species presence in that area.
Interesting. Yeah, we started off, Dive 365 also in the very beginning started off with some snails that were attached to a coral. They were pink iridescent ones. They were really pretty, and their bodies were this sort of pinkish red color. So really cool snails.
Is that a crinoid, a feather star?
It looks like that, yep. The pilots want to let you know, Daniela, if there's anything you want to see as we're cruising along, just give us a shout and we'll try to stop and zoom in.
Oh, thank you. Actually, I'm going to have to leave you soon. Got another link up,
Okay.
but I'd love to jump back on later, maybe this afternoon.
Yeah, definitely.
How long is this dive?
I don't think we have a set time right now. I think we're going to dive as long as we can into this afternoon. So we're usually on deck sometime around 5:30. So I would say it'll probably be at least done by then. But we've got a little bit of ground to cover, and then that last couple of hours is typically the very shallow stuff.
Mm-hmm. Well, that's maybe where I have more to contribute to. Things might be more familiar to me up there.
No, it's been great to chat with you. I think having these conversations really gets our audience at home an ability to learn who all of our different co-PIs are, but also us on the ship. So, we talk to Robin quite a bit, but it's quite nice to. Here's another Nautilus. Quite nice to meet all of you.
That's great. Yeah, likewise. It's amazing to have this opportunity to explore down here after having, like I don't know if you read what I wrote in the blog, but it's just this. There's this when the water is so clear, and you're surveying the shallow reef, but things are the reefs there are so steep in the coral sea often that you're drawn down. Your gaze is drawn downwards, and it's so hard to resist. You just really want to know what's down there, and that it's all linked together, right? So, whatever happens down deep in terms of even just currents and animals moving vertically up and down to feed or breed, it's all going to be linked up in some way, right? So there's going to be some sort of connectivity between what we're seeing in the shallows and what's happening down deep. So even though, like you were saying, the Nautilus have a very specific depth range preference, so do most other things, but they can overlap at various points, and where those overlaps might be, and what the exchanges might be during, in the places of overlap where species coexist. That's really interesting.
Yeah, definitely, and I think that relationship is something that I think different researchers have their specialty, right? But is always part of that scientific question of, how do all of the organisms sort of co-live together, but also how do they, who provides food for the other, and versa. And I think here in the deep too, we see a lot of that symbiosis of one organism being able to survive because another organism is there, and that's really interesting. So, we see a lot of the coral associates like squat lobsters and brittle stars and things that live on and around coral and vice versa. So it's really interesting.
Yeah. Yeah, and in terms of, this is a marine park, right? So the Coral Sea is managed by the Australian government as a huge marine park. And you know, we sort of at this point we're still in the open ocean. We're still figuring out how to set guidelines for protecting areas because there's so much we don't know about. And the more we can sort of reveal what's going on and how species interact, and especially what is dependent on other certain things. So what species are dependent on other species being there, all these links that we've just mentioned, they can give us a sense of, you know, what we really need to protect. What are the keystone things that hold everything together, and you know, where are the areas with most of the biodiversity? And so revealing, you know, these sorts of hotspots and where populations aggregate and where they spawn and feed, and this is crucial information when we're trying to protect marine ecosystems.
Yeah, absolutely. It's incredibly important, you know, this type of exploration and research and trying to figure out what's there. It's really hard, I think, for people to visualize and protect something that they haven't seen or don't understand. And so that's one of the main reasons why we explore, right? To look for and
Yes.
see and why, but also the really important aspect of being able to protect these environments from impacts in the future.
Yeah, and to be able to argue for their protection, because if we don't know, then there's very little. We have we have a thin platform to stand on when we're wanting to, you know, advocate for protection. So so this gives us a better foundation.
Yes.
Okay, thanks for having me on. I'm gonna pause and hopefully come back later and speak to you again, or speak to one of the other PIs. I look forward to seeing what comes up next.
Great. Thanks so much for stopping by, and we'll talk to you this afternoon. Thanks, Daniela.
Bye.
Bye. Looks like another fish. Has anybody been keeping count of the Nautilus this morning? There was a question. Yeah, I think we're up to 8. We saw 2 before we went live this morning. We had some issues with our live feed starting, so they've definitely been somewhere around 8 to 10 or so this morning. So definitely a rich Nautilus environment, as well as an entire field of old shells. So it was quite interesting. For those of you just joining us, this is Schmidt Ocean Institute Research Vessel Falkor, ROV SuBastian. You are seeing live images from the seafloor off the north side of Lihou Reef in the Coral Sea on the east coast of Australia. We have been out here mapping and diving for 5 weeks or so, I guess. And we are now on one of our last large reefs. And we've been doing some mapping over the last 2 days, and we found this really interesting sort of slump area on the north side, and where this is slightly protected from the wind, we were able to set up a dive for today. So thanks for joining us. Give us a shout out on YouTube and Facebook to let us know where you're from, where you're watching from. We have Chris flying the ROV, Ben's running the camera, Jimbo's managing all of the video annotation and collection, and I'm providing this stream to you at home, managing what you see. So if there's something else you'd like to see, let us know. Ben's co-piloting, sorry. I was corrected. He's monitoring our, absolutely, which is really important. He's also navigating where the vehicle is in relation to the ship at all times, which is really important. We don't want the two of those to come too close. So the ROV and the ship are always running in tandem, side by side, or front and aft, and that takes continual movements of the ship. And so Ben calls up to the bridge and asks them for a new move from time to time. So it looks like we have a group from the US and a group from some from Germany, and I think I saw the Netherlands and New Zealand, Denmark, Barbados, Newfoundland.
There you go. Oh, Jack Cavalli.
Ooh, look at this big fish. My gosh, is that the biggest one we've seen?
Jack Cavalli. He's one that gives me. Yep. Oh, they're aggressive.
It's checking us out, seeing what we're all about.
You're in their turf now. I'll fight him. I had one break my couple of my bites because they attacked. Get the 7 ready to put them on. Boys are salivating now.
Hi, Jason.
It's a pretty good size.
Close, yet still so far.
I just talked about that. Alright, no, check the one. Oh, yeah. Alright. Ready? Got some good video, Jimbo? Absolutely. Alright, we're up. This. Or it's because the current is getting wrong. Well, no. If I hit this, like trying to turn, is almost impossible sometimes. Keep moving it. Alright. Great, Cham, in your position.
Hi, Alexis. Nice to hear from you from Washington. Hi, Dad. Watching from the seawall. Aw. Must be a beautiful day in Rhode Island. Another one of these anemones in these moat areas. I was talking to Daniela about this. We saw the same similar, I don't know if behavior is the right thing, but environmental condition on the last dive where there's a bunch of sand sort of moved out of the way of this anemone sitting in the middle. Ooh, another little, I think we saw, ooh, some fish.
Let me get in front of them. Alright.
Interesting. There was a comment on YouTube. I saw tentacles of the anemone raking sand last time. Maybe that's how they're creating those moats. Yeah. So we just a quick shout out to our fellow crew members who are in Cairns, Australia, quarantining, so they can come and relieve us. Most of us on board have been here since January, if not before, and so we are hoping to have a crew change come up here in the end of this expedition in June, and so we have our relief are doing their 14 days of quarantine before joining Falkor. So just to give you guys an idea of where we are, I'll bring up the fleeter mouse here. So this view, you can see there's a sort of track along as we make our way to this wall. Our next geological feature is coming up in about 200 meters. There should be a bit of a rock that will climb up and over, and then a larger one. So this rock coming up is going to be about 25 meters high. And then the one after that is a little bit bigger, about 80. And then we'll hopefully be working our way up and along to this wall face of where this whole area has slumped and fallen off of the side of the reef. So thanks for joining us. This is ROV Dive 366.
That was a test. I was like, fish. Right, look at it. All right. Zoom me out for you. Zoom in. Tighten up, Jim.
Looks like a little fish on top of this rock here. There's been quite some colorful fish in this area. It's very iridescent, shiny, multiple colors.
There you go. Hold on. Stand here still.
another fish. Hope our fish army's watching. I haven't seen very many people on. We're just commenting on the geology here. It's very slab-like and very flat. It's very interesting, these cutouts and the steps. Ben, can you zoom in on the bottom there? It just seems like it's an interesting bit of rubble. We just zoom in, yeah, just zoom in on the rubbly bottom. It just has an interesting, trying to see if it's rocks or coral or... Very interesting. There's lots of little pieces of rubble. We just really want to thank everybody for joining us today on this ROV Dive 366. ROV SuBastian. Chris says hi, J-Rod. Ben says hi, J-Rod. No, he didn't really say hi. I said hi for him. And so we just want to thank you for coming along. We are live on YouTube and Facebook. All of our ROV Dives are broadcast live on both platforms as much as we can. We are currently working on the Visioning Coral Sea expedition. If you go to schmidtocean.org, you can learn more about ROV Falkor and ROV SuBastian. And this expedition. So our science team is ashore, working from their homes and institutions. And so Falkor right now is just running with crew members on board. So we have been mapping and ROV diving along these reefs in the Coral Sea on the Queensland Plateau for the last 5 weeks or so. He says hi back, Chris. It's really dark without the lights. The question on YouTube and Facebook. Chris is going to turn them all off here. So get ready for nighttime. Oh, look at that. You can see the lasers really good now. Creepy lights. Yeah, so it's 526 meters deep here. And temperature is 9.1 degrees Celsius. And it's pretty dark down here. We'll actually do that same exercise of turning on the lights off when we get up into the Mesophotic area. You can actually start seeing some light from the surface, and it's quite cool because it's all natural. It's a somewhat sunny, actually a fairly sunny day today outside. I should get out more. I spend a lot of time in this control room. So here's a quick image of our aft deck. As you can see, it's bright and sunny and not too bad for the weather. And here's a little peek inside the control room. So hello to everybody at home. You can see Chris is flying the vehicle. Ben is talking to the bridge right now about moving the ship. Tig is currently driving the ship up on the bridge. Looks like we have a couple of fish. I'm over here on the video station at the far end, and Jimbo is just off camera working on our station to the aft of the control room. We're following the fish I'm talking about now. That's this little one here. It's the one when I had the control room screen up.
Fish right there. You don't want to stop the photo. How about this one over here? I think they're both the same. He's like blue. He's different. The other one. More orange. He's got orange on top. They're both very, very similar colors. See the orange now. Hey, zooming in, Jim.
Yeah, there's a comment about where's the timestamp. I'm not sure. It was working when this system came back up today, and it seems to have stopped. So we had to reboot the computer that this runs on, and
Get ready, Jim. Here we go. That's great.
let me see if I can get it back.
You got it? Yeah. Alright, cool. I don't know what he's doing, but it's cool.
Sorry about that, everybody. I cropped it off. Yeah, it looks like we're just getting to the rock.
Okay, Jen, stand by. Spider coming up. Okay, zooming in halfway for now. Let them get in position.
So,
Once he gets in the middle, I'll zoom in tight. Okay, zooming in.
looks like a crab
There you go, Jen.
and a sponge in the background.
Okay. Got it. Alright. Something else.
So as you can see, we're just starting to sort of come into the side of this rock face. This is one of the many bits of debris that we're looking to go over top of in this area. So we are taking a look. It's an interesting geological feature. There's quite a bit of scalloping. We've seen this on other pieces of reef debris.
Stand by, Jen. Someone wants to come. It's gonna fly in the middle, Jen. Stand by. Alright. Got it? Go. Alright.
So there's a question from somebody on YouTube. Do you have any instruments for physically interacting with your surroundings on your ROV? Yes. So ROV SuBastian has 2 manipulator arms, and I can show you that view here. There you see. So there's a tool tray that slides out from underneath the bottom of the vehicle, and that allows us, when we are in expeditions that are doing sampling, we can actually use both of these arms to interact and pick up samples. We do not have a permit on this expedition to collect samples, and so we are purely an observer in this ocean right now. but we do have lots of different tools that can be put on and off of SuBastian and configured based on the expedition. So there's a question, what are the dark shapes in the background map? It's actually lack of data. So the black is typically means there's no points there. So this surface was made at a higher resolution so we could see these features. But what that means is that because of the way sonar works is that depending on where your track along an object was, your sound may not reach around an object. So even though we are on the surface and the sound is going down, a lot of times it's objects shadow that sound. So you may get one side of a feature and not the other, depending on which side the vessel is on. So we actually collected a little bit more data this morning. Right before this dive, we had a vessel track that did about this orientation. If you can see that blue line, and will be no more holes in that surface. We do have a smaller Saab Falcon ROV on board. For the question from, do we have any smaller observation ROVs? We do have one on board, but anybody familiar with that vehicle knows it needs pretty calm water and not much current and fairly shallow. So not conditions we find on these reefs. There's a big bamboo coral on the right. We're just discussing, the pilots and I, about whether they're going to fly over the rock. And Chris said he likes rocks and he's flying. So this is what we're looking at right now. I would say pilot's choice. And so we're going to take a look at this rock and eventually go up over it and then head on to the next one. But yes, this is a very long bamboo coral. Very pretty. Looks like a small crinoid maybe on the end of it. just put up the ROV pilot situational map, and this just gives you an idea of these ship and vehicle movements. So the Falkor is inside the red circle, and RV SuBastian is the yellow square, and we're headed for waypoint 2, sort of sideways. So we're going around the corner of this rock. We looked at it, we went up over the top of it, we came back down the front face of it, and now we're moving around it. So there's quite a bit of whip corals, bamboo corals, and other corals here. Very interesting geological structure that looks to be debris that would have fallen off the main reef structure. I'm not quite sure what you're referencing, Jules. The strings related to siphonophores, no. The long whip corals are corals, and are not in any way related to that siphonophore. Yeah, I think if you just skirt around the outside of it, then it'll be flat again, and we can work. The next rock is going to be bigger.
Yeah, so we just face of this and then around, and we're going to go back down the other side of the face, but the other clear.
Yeah. Hi, Les. Thanks for joining us. a pleasure to have you on today. Thanks for watching. One of the really amazing things about working on Falkor is meeting people from all around the world and getting to meet scientists and researchers from many different institutions. And so Dr. Les Watling was one of our former PIs, chief scientists on the Emperor Seamount expedition. And so I was able to sail with him and his team and do a similar expedition where we used ROV SuBastian to explore these amazing seamounts. So it's always fun to have people come back and say hi and keep in touch. And for their expertise when they come back in and let us know what we're looking at when we're here alone trying to identify the world. Thanks everybody on YouTube and Facebook for your shout outs for ideas of what things are. I know many of you are experts in your field and experts from watching hours and hours and hours of ROV diving. Thanks to the YouTube viewer who put on whip corals are more closely related to anemones, but they are in the same phylum as siphonophores, which is Nidaria. Thank you very much for correcting me on that. A very interesting fact.
Big fish.
Chris has spotted a fish, which I think you see in the center left of your screen.
I'll go in there.
This one's different. Maybe. I don't know. I'm not a fish person. It's interesting though. Just going to bring up this inset map again so that you can see where we've made it, and also the view from the sit camera. So the sit camera is looking up and we're looking down in the science camera, and so you get sort of the dual perspective of what this rock rubble looks like. On the multi-beam data you can see that there are these definite debris rubble fields from this huge area that has fallen off the side of this reef key.
We don't see any more fish the rest of the time.
Hi Kai, thanks for joining us. Hi Jasmine, thanks for saying hi. Jimbo Jasmine says hi. Just a quick heads up everybody on YouTube, it's a family friendly show, so I will hide your comments if profanity is used in it. It's an automatic filter on YouTube for us, so if you would like your Comments shown, please don't use swear words.
Hello, Debs. Can you hear me? This is Robin Beeman calling in from Cairns, Australia.
Sorry, Rob, are you there?
Yes, Debs. I'm just calling in for the first time today. This is Dr. Robin Beeman from James Cook University, based in Cairns, Australia. And speaking to you, I'm about 650 kilometres due west of where you are now.
Great, thanks for joining us this morning. Sorry, I was just off comms for a little while trying to set up my own camera. But yeah, go ahead and if you want to talk to the audience a little bit about why we picked this dive site. And we're just on the rubble in between the big, we're headed for the big rock now. And here's the...
Yeah, thanks, Debs. Oh, that's great. Okay, so over the last few days, the Falkor's been mapping around this Lihou Reef. This is the largest of all of the Queensland Plateau reefs. It's nearly 100 kilometres long, about 34 kilometres wide at its widest, and Lihou Reef ranks up in the top 20 largest atolls in the world. So it's a very, very big shallow coral reef system, but when we say shallow, that's only the bits that have been previously mapped. And what Falkor has been working on over the last few days is mapping all around the deeper flanks. Now, all of these 30 or so large reefs and banks on the Queensland Plateau are really big edifices. In some cases, they're over 1 kilometre high, right? So the surrounding Queensland Plateau surface averages around 1,000 metres deep, so 1 kilometre deep. And then we have these very large reefs that rise up from the depth. And so hence the Falkor has been really focused on mapping the edges of these reefs, the deeper flanks, of which we had really no idea, no understanding at all of what existed down there. And so it's been a real thrill to see the detailed 3D maps being revealed through the multi-beam mapping. This is this modern sonar technology. Very detailed, in fact. We can see individual boulders and the like. Generally, a resolution of around 5 metre resolution, so we can resolve features down to the scale of my bedroom where I'm speaking from, which is quite remarkable. And then once we have those really detailed 3D pictures, we can actually start looking for sites. Now, one of the big factors that's affecting the ship out there at the moment is the weather. The weather has been a bit ordinary, quite windy. It's up around 20 knots. And that's, while it doesn't limit the ship so much from a mapping perspective, although it can be uncomfortable when you're dealing with swell and waves, from ROV perspective, you really need a nice sheltered spot. And so yesterday afternoon, I sat down, virtually, of course, with Debs on the ship, myself here in Cairns, and my other colleague, Dr. Jodie Webster from the University of Sydney. And we looked through the 3D maps, and we came across this site. It's at the very sort of northeastern end of Lihou Reef. It has a base around about 600 metres, where it starts to flatten out onto the plateau surface itself. It looks like we've got a nice spider crab in front of us. I'll just stop for a moment to talk about the spider crab. If we've got the laser beams, I think they're on, yes. Yep, in the bottom centre of the view. And so this is a good 20 centimetre across spider crab just sitting on the seafloor. Maybe 30 centimetres across. This would probably make a nice meal if you were to catch it. So we decided on the site because it had a very, very obvious undersea landslide had occurred, where the entire face of the steeper slope had sheared away And if you have a look at the inset map on the bottom right, you can see this very distinct scarp face. It's got a bit of shadowing on it. This helps with the human eye to see where bumps and lumps are on the seafloor. But you can in fact see that that stretch that's collapsed. Now, Debs, can you just tell us about how wide is this landslide collapse feature?
About 2.9 kilometers.
Yeah, thanks. So we're talking about a 3-kilometre chunk of the shallow reef from the top, the shallower parts, all the way down to the bottom, so over 500 metres now, has just given way. And you can see the steep scarp behind. So imagine a big, an area of, imagine a large cliff that's just completely fallen away. And so what you're left with is a series of blocks and debris fields at the base. So we've chosen this ROV dive to run along the plateau surface, which is where I am now, over a whole series of blocks, some of which are quite small and some are much larger. And then the idea is through the day we will run up along the side of that very exposed scarp face on the right-hand side, and then we'll traverse across the face itself. And then ultimately we'll come all the way up into the shallower waters as high as we can, according to how safe it is, because the Falkor is sitting quite close to a shallow reef at the moment. Yeah, so right now we're actually looking at lots of boulders. Imagine this is just part of the big debris field that's collapsed, and now it's lying scattered at the base, at the foot of this slope. And I'm pleased to see that that Kai T has joined us. Kai is one of our other principal investigators, our sharp-eyed ichthyologist. Kai is at the University of Sydney, and when we were working up in the shallow Mesophotic zone from a previous dive, he identified new fish species. So it's been really great to have Kai and his colleagues joining us from all around the world. So good to see you here, Kai. Perhaps later in the dive, as we get up into the Mesophotic zone, that warmer sunlit area, you can actually join in audio with us, and we can see what fish we can find.
Okay, and I just wanted to introduce everybody at home. They've been hearing your voice for weeks, and so I just wanted to give them a quick sneak peek of you. So say hi to the world.
Yeah. Hello, everyone. Yeah, this is quite an unusual expedition. Debs has already explained the difficulty that came up that initiated this whole expedition, where we were marooned on shore, so to speak. The Falkor had to go out and do its expedition without us, and so we've had to figure out a way of keeping in close contact. And it's been difficult, but it's been a lot of fun. And probably the most fun is actually speaking here from my bedroom, my spare bedroom here in Cairns, to the audio, and then which is streamed live to the world. Of course, I've got my family here as well. They had to be homeschooled for a long time, and my wife was trying to run a business from here. So it was all really interesting. But I was able to bring them in at times and say, hey, check this out. Oh, they're seeing a Nautilus live. It was, not your usual COVID stay-at-home experience. So anyway, we've managed to get through this as best we can, and that has been a lot of fun. But it's not the same as actually being on board a ship. And as a long-time ocean researcher, I like going to sea, and you can actually be fully involved with all the shipboard operations. Of course, it's a lot more difficult when you're on land. But anyway, it has been a really remarkable, really great experience.
Yes, for us as well on Falkor, you know, it's a very different expedition not having all of the science team here with us and doing all of this remote telepresence work. So I'm glad that we were able to work through all of the technical challenges we've battled along the way and bring you guys some audio and narration from our expert science team ashore and still be able to map and live dive with the ROV for all of you to watch at home.
Yes, Debs, you know, huge credit to the Falkor and the team to be able to overcome these quite challenging communication issues. I mean, one of the things that I need to do as chief scientist is keep a fairly good situational awareness of what the ship is doing. And that way, you know, that way we can actually bring some, you know, we can help guide where the ship is going. It's all about forward planning and dealing with, you know, we're often talking about plan A, plan B, plan C. And this is important because it's quite a dynamic situation and of course the weather plays a big factor. But we're going into areas that are really unmapped and we don't know what's there. And so with the Falkor going into these places for the first time, we're seeing these pictures, the 3D pictures and of course the ROV pictures for the very, very first time. And so it's important for us on shore to be able to really keep connected with what's been happening. And so we do this in a number of ways. We share a screen. We use TeamViewer. We have a direct picture of the operations room monitor, so I can see exactly what is being mapped in real time. And sometimes things pop up and I can actually jump on and say, just with you know, a chat or audio, and say, hey Debs, you know, what's going on here? And we can quickly jump on and talk to each other, even though we're hundreds of kilometres away. We often do Zoom meetings. We do them every day. We have a morning Zoom meeting that allows me to talk directly with the ship's master and all the rest of the management team on board the ship. The other thing we do is from my side on the shore, I then have to distribute that information out to all the other principal investigators so that they are involved, they feel like they're part of the expedition as well. So there's a huge amount of communication, both ship shore and also happening offshore-wise, to make this work. I guess the other really interesting thing is, as we've expanded up our number of principal investigators of the voyage, we can bring more and more people speaking in, more experts speaking in on these ROVs, these ROV dives. And so I think on our last one, that was 365 up at Diamond Islets, we had, you know, 4 or 5 principal investigators joining in through that dive. And that's been a really great thrill to see that evolve.
Yeah, absolutely. It's been great to have, you know, the continued audio commentary and just a perspective from everybody in their different specialty areas.
Okay, if I put my geology hat on at the moment, I'd like to, we're just talking about the boulders and blocks. So it does look like a great big scree slope, and that's exactly what it is, but it's on a very big scale. So Debs pointed out earlier, it was like a 3 kilometre long chunk of the reef has collapsed. And so what we're looking at is this jumbled pile of boulders, all piled up on each other. And it's pretty big scale too. If you can imagine, you know, something on land, like a 3 kilometre stretch of a mountain that's around, let's say, 0.5 kilometre high has collapsed. So that it would obviously result in a lot of material deposited downslope. And that's what we're flying up at the moment. The other thing, I guess, what we hope to see in this, the exposed scarp face is the layering that we expect is the build-up that's created these reefs. Now, all these reefs are very old. They're Miocene to Pliocene in age. They've been built up on the upturned continental blocks These continental blocks are just pieces of Australian rock that was sheared off part of that breakup of Gondwana many years, millions of years ago. And these reefs have basically built up on the corners of those blocks. And we know this from deep seismic work that was done in the late 80s, probably 1970s and 1980s, where, seismic data, it's almost like looking at a slice of the earth. And you can actually see these coral reefs have grown up from these big continental blocks. But when I say grown up, we're talking about limestone here. So coral reefs that have built up layer upon layer. And as the plateau itself, the Queensland Plateau, has subsided through time, then as long as these reefs have been able to stay close to growing near the sea surface, then they continue to add another layer. Ultimately, you end up with this sort of big wet cake layer effect, each layer building up in time. So you end up with a coral reef that's nearly a kilometre high in places. The living part of it is only at the very, very narrow upper surface within that photic zone. And so, of course, by layering, the deeper parts are older. The shallower parts are younger. I would think that the rocks we're currently flying over are probably millions of years old, but it is carbonate limestone. Limestone made generally by coral and algae. And so while we can't do any sampling on this expedition, like physical sampling, because we don't yet have permits for it. We don't have permits for that with this expedition. In a expedition coming up, we will be able to collect these rocks, and then we can do all kinds of clever things with them, like age dating and looking at their isotope analysis, and that can tell us a lot about the environment in which these limestone rocks were formed.
Thanks, Rob, for that explanation. I was just discussing with the pilots how we were going to approach this rock face. So they're going to do some investigation around it, actually, go fly up towards the top and then come back down and then maybe fly around the eastern side of it. So we're working on coming up over this boulder here right now, and the ship is positioned to our east.
This is really a very prominent boulder. It's probably the biggest one that we'll go over today. It doesn't look like just one complete boulder. It looks like a big jumbled mass. Just curious, sort of what height are we talking about to go from the bottom of that boulder to the top? I'm guessing it's probably around 50 meters height.
Yeah, it's about 60 meters high, maybe a little bit more. So yeah, Chris is going to work up to the top, and then, but he's going to start at the bottom.
I think I just saw a Tina 4 drift past.
Yeah, there's been. Sorry, I was going to say there's been a lot of biological in the water, both out on the plain area and more flat area, and here as well. So there's been quite a bit of life swimming in the water column.
Okay, yeah, we don't talk so much about that a lot because the ROV tends to sit closer to the seafloor, but it's still very exciting. We've seen all kinds of things before. I mean, your previous expedition on the Ningaloo Canyons, you came across a giant siphonophore, huge jelly-like medusae as well. Here out in the Coral Sea, this particular part of the Lihou Reef, it's at the very southeastern end of the Queensland Plateau, and it's fully exposed to the Coral Sea currents that sweep through there. There's really no real landmass east of us, except perhaps Vanuatu, which is thousands of kilometres to the east. So there's no barrier here. Hence, all the waters here are fully oceanic. In this particular zone, this depth zone, it's subtropical mode water, so it's cooler. But as we're climbing up on this transect, we're going to be crossing through this thermocline. Seems to be very distinct thermocline. We see it in all the other dives. It's around this 500 to 450 metre mark. So I'm curious as to what the water temperature is at this 494 metres.
9.6 degrees Celsius.
Yeah, okay, so it's definitely warming up. Like we were at a dive the other day, they were down around 900 metres and it was just over 5 degrees. So, here it's starting to warm up. So this is actually we're actually at the bottom of the thermocline, and it's probably around 50 to 100 metres thick. But once we pass through it, we'll be in the Mesophotic zone. Yeah, we can see these comatulid crinoids sitting on the rock. They've got these very distinct cirri, or legs is probably, I think cirri is the correct term. They can move. They're fully mobile. Certainly in the shallow coral reefs, you see a lot of these, more so at night time, and they're often found sitting up on shallow coral reefs, waving their feathery arms in the currents, collecting food. Down here, they're just using the rock as a substrate. What I've really enjoyed is seeing the stalked crinoids. These are sea lilies, really a relict fossil, relict animal in these deeper waters. We haven't seen any, I don't think, on this particular ROV, but certainly in some of the deeper ROV transects we've done the Coral Sea, we've found lots of these sea lilies attached. Yeah, so one of the other features we've found in this thermocline is it isn't actually, it tends to be a little bit barren, not barren to the point of no animal life. There's still life there. We just have to look a bit harder for it. And I think that with the thermocline, it is this rapid rate of temperature change. I mentioned it earlier. It's that point where it's too warm for the cooler water animals to migrate into and survive, and it's too cold for the warmer animals that live in the Mesophotic to I guess push down within. And so a thermocline is never a distinct layer itself. It's moving through time because we're talking about a water mass, and so we're looking at a sort of a layer probably about 50 to 100 meters thick, and we're just kind of passing through it now. So we're still seeing cold water corals, though. There's looks like a dead scleractinian coral attached underneath these ledges. Now, ledges are important. We found on all of our dives that ledges, because they're oversteepened, there's not a lot of sediment accumulating, and so they're great little places to stick your head in. This is what I love about the SuBastian. This is a very large piece of kit. It's 3.5 tons. It's the size of a large car, and yet it can just kind of poke its head up under these little ledges and into nooks and crannies and caves. So it's quite a thrill to be able to do this. And you can actually see. So this is a dead scleractinian coral, but it's got things attached to it, look like little feather stars and the like. Oh yes, with the light we can see a lot more. So these these long strands. These are probably bamboo corals, and they've got little crinoids, feather stars attached to them. Yeah, again, just trying to. There seems to be a feature they push further out into the water column, and so they're just using whatever structure they can get to do that. And in case in this case, they're just using other long corals to attach to hang on to. And the other thing I noticed is just a very faint staining on the rock. You can see this sort of darker brown colour. This is likely ferromanganese oxide. It's more prominent as you get deeper into the water column. So I remember down at our first dive at Osprey Reef, it was down around 1,600 metres, 1.6 kilometres deep, and any exposed rock was almost jet black. And this is the ferromanganese oxide. We will lose it as we push through this thermocline and get into the Mesophotic zone. Yeah, Deb's had a question. What is the surface temperature at the moment?
It's 25.7 degrees.
Got it? Yeah, I come to a screenshot.
Yeah, so I would be quite comfortable swimming there. I'm a local. I tend to do most of my local Cairns person. I tend to do most of my diving and snorkeling on the Great Barrier Reef at summertime, and you can get temperatures up around 30 degrees Celsius. So it's almost like swimming in bath water. And we just use a very thin wetsuit, more not for the cold, but really just to stop from getting sunburnt. In fact, I had one of my great diving experiences this last Christmas gone. My son had just turned 12 and he got himself a junior PADI dive ticket, and I accompanied him out on a liverboard dive boat out in the Great Barrier Reef. And we went out of Cairns. We spent 3 days diving together, myself and my son. A beautiful coral. I remember this area, a forest of sea whips, and we were just floating over it. It was a really, really great experience to go out with my family for the first time, because I've been scuba diving in the Great Barrier for over 30 years, but that was quite a thrill to go out with my family. Hi, I hope you're watching from ashore with your fish army. I wonder if you could ID this small fish we've picked up with the large eye. Hey Debs, it looks like our fish army are in full flight here. That's good. We got Epigonus, cardinal fish. This is great, and it looks like Tiffany C is watching. And welcome aboard, Tiffany. Tiffany did her PhD on Mesophotic fish on the Great Barrier Reef, and I was privileged to be on some papers that she developed. She worked with mainly baited remote underwater video systems, ROVs, and really got probably the first really comprehensive picture of the population and species of these mesophotic fish right on the shelf edge of the Great Barrier Reef. So she has quite a great understanding of these deep water fishes too. But what I'd like to do now is just bring Richard Fitzpatrick in to do some audio. In fact, Richard is here in my spare bedroom in Cairns. He lives here as well. It's the first time he's visited me during the whole COVID crisis. But Richard's actually doing some filming of me talking here. But I thought I might just bring him on. He can just jump on the headset and perhaps talk about the Nautilus that we saw very early in the YouTube stream.
Hi guys, Richard Fitzpatrick here. We had to move the mouse because Rob's a southpaw. Had to get over into the right-hand side. But yeah, first time I've seen Rob since COVID started, so we're allowed to move around a bit more freely in Queensland at the moment, and so I've taken the opportunity to pop in and get some footage of Rob talking virtually on this whole experience, which is pretty amazing that everyone can work from home. And see Kai's online. Hello, Kai. It's been a while since we had Mexican food up with Lyle Squire at Cairns Marine. So yeah, no, this is great. But the Nautilus this morning, that's what Rob wanted me to jump on, talk about is, we used to keep Nautilus in captivity when I was at Manly Aquarium back in the day, which sadly closed last year after 50 years of being open. But with the Nautilus, what we do in captivity is we actually keep them in a reverse day and night cycle. So during the daytime, they're basically resting. So at nighttime, they're doing their vertical migration up into the shallower waters to feed. And so what we came across this morning was obviously a Nautilus in the daytime doing what they normally do, which is pretty well nothing. And it was actually, it looked like it was fully asleep. So the hood was pulled down, which is that big leathery top on the top of the animal's head. The pinhole eyes were pretty closed up. The tentacles were sort of tucked in, and that's basically how they sleep. In captivity, they would just hang on to something, like a bit onto the glass or onto a rock, and just shut down. And so then at night time, they'll come up and start feeding on things like crustaceans and all that kind of stuff. So the other interesting thing with them in captivity is when you ever see a Nautilus in captivity in an aquarium around the world, there's quite a few aquariums that have them, is that you'll see the black line forming on the shelf from basically when they were caught. And then from the growth on the shell, as it increases size in the spiral, that there will be black markings on it. And we used to call that the stress line. It's not that the animals are overly stressed, because they do feed and they do mate very well, and they're always mating when you've got them in captivity. But it probably has something to do with how the shell is forming, that they're no longer under such a great deal of pressure. So the thing about the Nautilus is that it's a bit unusual to the other cephalopods, the group that it belongs to, like the cuttlefish and squid and octopus, where the Nautilus are actually quite long-lived, so you can get quite a few years out of them. Probably because they've got a slow metabolism and they don't move around much like their counterparts. But all the cephalopods have a copper-based blood, unlike us, which is iron-based, but the copper has a less affinity to oxygen. And so with the cephalopods, they tend to have a higher respiratory rate to get that blood moving around, to get the oxygen they need. And indeed, most of them will have a heart on the top of each gill and then a primary heart in the middle. So 3 hearts, green blood, pretty well an alien, and pretty amazing critters. So yeah, it's been wonderful seeing Nautilus in the wild on all these dives and the different locations. And so many, yeah. It's been cool, hasn't it, Rob? I know Dr. Andy Dunstan, who's done most of the work on these in the Coral Sea, has been pretty excited about it all. And what's the sea star there? Yeah.
Richard, we have a question.
a site, and there they are.
Yep.
We have a question for you from the control
Good.
room. Earlier in the dive today, I'm not sure if you guys saw it, but we came across what was like a whole field of dead shells. So it was quite the most we've seen on any of our dives, and it was quite large, and I don't think we could really count how many there were. So the question was, do we know why that is? Is it just happens to be something that's coming down off of higher, or is that an aggregation of some group dying, or predators, that kind of thing?
That's interesting. Now, I didn't see that footage. I mean, off the top of my head, when they die, the shells tend to float as the animal's sort of decomposing, and then they slowly take on water and go down. So if you had an aggregation like that, I'd probably think it's more to do with the ocean currents.
Interesting.
Like an eddy or something forming that's accumulating them in one spot rather than a mass death die-off in one location.
Right.
I think it'd be more oceanographic than biological off the top of my head.
Yeah,
But
Daniela.
yeah, I'll definitely have to go back and have a look at that vision. And
Daniela was on with us at the time, and that, I think, was her hypothesis as well, was it something related to current. So we were discussing, she was talking about how these reefs here on Lihou are a lot less protected than others from more dynamic flow, both because of the size of the reef internally, and then also because of the more openness to the deeper water around the north and south sides.
yeah, no, for sure. Well, there's some more fish there. Kai will be getting very excited. I do know for Andy Dunstan's mark recapture work at Osprey Reef that shells were recovered on Cape York. So obviously the animals hadn't swam all that way, but after probably natural mortality, and they floated, that they've been dispersed. So he's had mark recaptured shells sent to him from quite a few locations in Queensland. So we do know the shells go a long way. And of course there was some pretty famous research done at JCU years ago about left-handed and right-handed thongs, or what we call them thongs, Americans call them flip-flops, in looking at how they were washing up on beaches, whether they were left-handed or right-handed. And I can't remember the guy's name who did the research. It was around our time. Rob and myself did undergrad together, actually. Carlos, but yeah, it was an amazing bit of work he did. But looking at predominant currents by looking at the left-handed or right-handed flip-flops washing up on beaches.
Interesting.
And of course, you have other left-handed and right-handed animals out there, like the blue bottle, or what you guys call Portuguese man of war. Half of them will have a 45-degree tilt on their sail to the left, and others will have 45. The other half population will have 45 to the right, which means in prevailing wind, the whole population won't get wiped out at one go. So they go two different directions.
Interesting. I never
That's
knew
nice.
that about
Crinoids.
those. Couple of fish here.
Rob's putting out. Prasinga.
Prasinga.
Prasinga sea stars. Wow.
Yeah.
See, Kai was sending me some messages. Where were they? They don't. Kai's got a photo of him holding an Emmy Award. Well done, Kai. That's beautiful. Okay, Kai, what's that one? Zoom, please.
We did. We zoomed. You should get it about now.
Antheus. Yep, well, there's one of the deep water Antheus. Yep. Nice. Of course, I'm here to look for sharks, so more sharks, please, guys.
Jimbo, just so you guys know, I'm giggling because Jimbo just directed that fish from the ship. He goes, "Can you zoom in, please? And now can you turn the fish around?" And the fish absolutely turned 180 degrees. So that was a pretty funny moment here in the control room.
Well done. You guys are doing natural history, yeah. Tiffany's just said by the wind sailors also have the left and right-handed variety. Yep. So that's the Valella.
Interesting.
Also do that. So, but they're all in that same group where they, amazing animals. Yeah, beautiful. And there's talking about jellies. There is a oceanic box jellyfish that we find out at Osprey, and it's also now been worked out it's the same one that does occur in mass blooms off Waikiki. And out on Osprey, a few nights after the full moons during the summer months, you can get mass aggregations of these, which are pretty amazing. So yeah, it's cool stuff in the shallows.
Just zooming in, we've seen quite a few snails on this dive today, and so been quite interesting. There was a question on Facebook: can Nautilus reproduce in captivity? Do you know if you know the answer to that? Oh, sorry, I didn't know if that had already been answered.
They can, they can, and they do. I actually watched one hatch out at Waikiki Aquarium. So Waikiki Aquarium and the aquarium in New Caledonia were the leaders in Nautilus in captivity back in the beginning. Yeah, so it's amazing. And when they hatch out, I mean, the egg looks like a little alien, weird shape. But when they pop out, they're an absolute tiny replica of adult Nautilus. So the full little spiral and very cute, about the size of your thumbnail. But yes, they do reproduce in captivity. The one thing that took a while for people to get their handle on was that the eggs took a long, long time to hatch. So when people thought, oh yeah, something's wrong, so yeah, but I can't remember the exact period, but it was a very long period of time. That was Bruce Carlson's work in his team in Waikiki back in the day. Yeah, it was pioneering. It's like what's happening now, isn't it, Rob?
Yeah, it seems like Kai's
it's bizarre
saying,
to see someone else in 3D at the moment, I must say.
Kai's saying that last fish maybe is undescribed, so it'll be interesting.
And when I talked to Rob, there's no 20-second delay in the response. This is bizarre.
Yeah. It gets even worse if you watch it on YouTube.
Got it?
Yeah.
Okay, I can. Hold on one second. Okay, here you guys go at home. There's Richard and Rob in his spare bedroom in Rob's house. That's where they're narrating from.
Hello from Cairns.
Is that how you're supposed to say it? Cairns?
Yes.
Like with a long, drawn-out
Pretty cool.
Cairns accent?
Cairns. Yeah, just like you want a lot of beer cans.
Beer cans. Oh.
So yes. Sandy Lee also jumped on saying it is illegal to capture Nautilus anywhere in Australia. And there is no one can capture marine life in the Coral Sea marine parks unless it's under strict permits. So yeah, luckily our Nautilus population is very well protected here, particularly now that we have the Coral Sea Marine Park declared, which was a long drawn-out process. And there's lots of people, lots of biologists, and everyone getting involved, talking about things like the Nautilus, the sharks, and everything to get it protected. And when it finally came about, it was an amazing effort. But of course, Nautilus shells are under a great deal of pressure in the Indo-Pacific region, where people, they're still collected for the shell market. So it's one of those things, as the conservation groups say, if the buying stops, the killing will stop too. So yeah, please don't buy these kind of shells, because it just leads to these amazing animals getting captured. And until Andy Dunson was doing the population work on them in the Coral Sea, we had no handle on what the population was, the age structure was of the stocks, and all that kind of stuff. So they're extremely vulnerable to fishing pressure. Oh, here comes another one of those fish.
Oh, it's a coffin fish.
Yeah, grumpy coffin fish.
Yeah, grumpy fish.
That's what happens when I haven't had coffee in the morning.
It's yellow. It's really pretty. We usually see them orange.
And that red underneath is really nice too. A little one. Chinax, yes.
Trying to sit down on this rock face is not easy.
What a face. Last time I saw a face like that was when Kai had Mexican food up here with us when he was visiting on a trip. He said he could handle the hot food, but he couldn't. Oh, look at that. That's one of the best colours in that one. The best coffin fish so far. But you can see how the pelvic fins and the pectoral fins are almost modified as little feet so they can walk along. Beautiful, guys. Yeah, about 10 centimeters across.
Yeah, I think just a little less than 10 centimeters.
Kai's sending messages through about the nightmares he still has about the Mexican food that night in Cairns. Oh, what's that? It's nice when you're looking down the side of the wall like that too. The whole depth. Might jump you back on for a sec. Okay, guys, I'm going to jump off and I'll put Rob back on. I'm going to keep filming. Richard out.
Okay, thanks.
Oh, cup coral. Thanks, mate. Yeah, thanks to Richard. He actually came in to just film us doing this. Well, we've been doing it for a month now, recording and speaking from our respective temporary offices around the country. So the other principal investigators, it's probably good time to introduce them. So there's Dr. Brendan Brook from Geoscience Australia. He's based in Canberra. We've got Associate Professor Jodie Webster, also University of Sydney. Kai T, I mentioned earlier, our ichthyologist at University of Sydney. Dr. Tom Bridge from the Museum of Tropical Queensland in Townsville. He's the Senior Curator of Corals there. We have Dr. Merrick Eakins, our deepwater sponge expert from the Queensland Museum in Brisbane. Richard, who you've just met, he's based here in Cairns, obviously. He's with BioPixel. So BioPixel, a long-time documentary maker in the Great Barrier Reef and Coral Sea. So you gather Richard's got a lot of experience out in the ocean. And Dr. Daniela Ceccarelli from Coral Sea Foundation and based in Townsville in Queensland. And so at times we've been able to bring all of these people on to speak, and we really also appreciate all of the experts ashore who are watching in on these dives and helping us ID animals. It's very welcome.
Sorry guys, we're just zooming in on a few of these things again. We just did a lunch handover, and so I'm now doing Jimbo and my job, so just it's a bit distracting trying to remember to take frame grabs while reading comments and all the rest of it. So anyway, we got to look at the orange thing and the fish, which I think is an anemone, and that beautiful feather star, I think, or coral.
So just
So I'm,
through your knowledge,
yeah.
I would say that this amount of 75 to 80 meters tall.
Okay, so we thought about that 80 meters or so earlier. So the Chris is saying the mound is about 70 to 80 meters tall. So we're just
Right now
gonna.
I'm 70 from where I started.
Okay. And the water temperature's increased a little bit. It's 10.6 degrees Celsius. So we're about halfway up this, maybe a little more than halfway up. At the waypoint 4 on your screen, Chris, is the peak. So it kind of is a bit of a slope here to the top.
So once I get to the top here real fast, I'm going to start heading back down to the base.
75 meters, okay.
Back up and go back down.
Okay, so there's some questions that's come in on Facebook. What kind of data is being collected on this dive? So along with the video data from both the Science Cam and Sit Cam. Hold on one second. I'm going to take some frame grabs. Sort of like chewing gum and walking at the same time. It's really hard. We also collect CTD, so there's a CTD and a dissolved oxygen sensor on the vehicle. So we collect all of that as well as all the navigation data. And so that gets all logged. We also do an event logging system. So this tells us who is piloting, co-piloting, video annotating, and then also allows us to make notes within a database of when we saw things. Thanks, Dad, for answering the people at home. Meters to feet. We work in meters in the oceanographic world. Google helps to do that conversion. We just had a pilot change. So Russ is now flying the vehicle. human impacts and we have seen lots of human impact on other expeditions in other places, so I'm not sure if it matters distance-wise. And then, Dan, your question about animal fossils, I'll have to wait for Rob to come back to answer. I'm not sure. So hold on to your questions. Our chief scientist should be back in a little while, and we will try to get those answered. If anybody on YouTube and Facebook knows, go ahead and shout them out.
afternoon, everybody. You're watching live video from ROV SuBastian, deployed from research vessel Falkor. We're currently diving on the Queensland Plateau off eastern Australia, and looking at a rock covered in crinoids, a little bit of coral. Water temperature is currently 11.6 degrees centigrade. There's a nice sea star on the right hand side of the picture there as well. We're going to be slowly moving our way up the reef towards the Mesophotic zone, where we expect things to change. You'll get more of the shallow water corals and probably a lot more colourful fish. So keep watching. Hello, Daniela. You should be live now. In theory.
Hi, just testing whether my audio works.
Yep, I think you're live on the stream now, Daniela.
Great, thank you. Can you hear me, Kai?
Hey, good afternoon, Danielle. Yeah, I can hear you very well. How are you?
Great, great. Good to finally get to chat.
Sorry, I'm freezing at the moment. It's so cold in Sydney.
Oh, well, I'm on Magnetic Island, which is sort of similar latitude to Lihou Reef, where we are underwater right now. So it's quite nice here.
Yeah, lots of interesting things today going on in the dive. I can see a lot of people on the YouTube chat commenting and giving, really useful and valuable ideas. So thank you guys for that. Just a shout out here, Jason and Ellison, Joe, and all our usual watchers on YouTube.
Kai, could you explain the difference between a goosefish and a coffin fish?
Yeah, sorry. Yeah, I think they're from different families. So goosefishes are from the family Lophiidae. I'm not sure what Chonyx is, coffin fishes. Let me just have a quick chat. Yeah, they're from a different family. They're from Chonacidae. I'm not super sure what the morphology is in terms of differences. I'm not very familiar with these groups, but they're from totally different families. And I think the general appearance, kind of the bottom dwelling, modified pelvic fin, pectoral fin appearance is just a convergence between both families of fishes.
Okay, because I thought what we were seeing was a coffin fish, but of course I'm not a fish expert, and people on YouTube are saying goosefish, so I was just a bit confused. I thought,
Yeah, the one that we just
thing?
saw is a goosefish.
Ah, right, so it was a goosefish.
Yeah.
Okay, so I noticed the goosefish has definitely got the lure then, and I don't think the coffin fish has a lure. Is that correct?
He's still there. He's been a bit mad.
Yeah, you're right. I don't think coffin fishes had the little lure at the top of their head.
That's great. Okay, that's just my curiosity there. So thanks very much for clearing that up. I'll let you and Daniel get back to your conversation. Sorry about that.
No worries. No, this is a group of fishes that I'm not very familiar with as well. It's really good to have all these people commenting and giving really useful feedback and identification, because I'm also learning a lot with some of the deeper water fishes here that are kind of beyond the Mesophotic realm.
Yeah, it's been really great to see everyone jump on board and help out with IDs and things like that, because I don't know, like you, I consider myself a fish nerd, but down here I feel like I don't know anything. I mean, you sort of do see, parallels with how things are built and how they behave, but obviously they're adapted to very, very different conditions.
Yeah, I am totally on you, with you on that one. A lot of really interesting fishes here that we're seeing. A lot of them are completely unfamiliar to me. It's, really good. We're at kind of like the 400-meter mark now, where we're getting a bit of mesophotic species start to trickle in. Things from the family Serenidae, for example. We've seen Plectrantheus and some unknown Anthiadine Serenids. In that regard, this is kind of a more familiar ichthyofauna of the Mesophotic, but you're still getting things like epigonas, coffin fishes, goose fishes, these kind of things, and Antigonia, that really nice boarfish that we just saw. They're kind of more characteristic of deeper reefs, kind of in the 500-meter and below realm. We don't really see that very often in the Mesophotic area. So now is kind of a good area and depth where we can see both kinds of fishes mixing together.
Yeah, and I'm also curious to see what you think about this. What parallels are there between the sorts of fish with the kinds of adaptations we see in the deep sea and temperate species that we might find at shallower depths?
Are you talking in terms of ecology or morphology here, Daniela?
Well, a bit of both, and also taxonomy. Whether you sort of tend to find the same families, say, that are sort of depth specialists, that are also then found in more temperate waters. That there's more parallel in the taxonomy between the deep and the temperate than there is between the deep and the shallow at the same latitude.
I think we see more common taxonomy between shallow and deep reefs as opposed to temperate reefs versus deeper coral reefs. There's definitely a very big difference in terms of the kinds of fishes and the kinds of species you can find that are suited for temperate regions. I think in that regard, we're looking more in terms of biogeography and ecology rather than the taxonomic distributions of fishes across a kind of a niche going from shallow to deep. Although one of the more interesting families of fishes that we've also seen in the last few dives is Calanthiidae, which comprises several genera, including Calanthius, which is very predominantly represented in temperate regions, like in southern New South Wales, New Zealand. The other genus in this family is Grammatinotis, but they're restricted to very deep reefs across all tropical and temperate latitudes. So you see a kind of distribution in terms of genera based here on tropical latitudes, but in very deep reefs. And then the sister genera being restricted to shallow water temperate reefs. But I would say by and large, most distributions of fishes follow, in terms of taxonomy, distribution in parts of depths on the same reef rather than temperate and warm water ecosystem. If that makes sense. It was a bit convoluted there. I think I was going off on a tangent.
No, that was really interesting, and you know, thanks for the answer, because it's really sort of hard to know when you're not familiar with the deep sea environment. And in fact, you know, we're exploring things we've never seen before, I guess, down here. And so I guess just to make sure I understand, what you're saying is you find more similarities within the same latitude than within the same sort of temperature depth range.
Yeah, in general, I guess there was a kind of point that I was trying to make and trying to get across here. Yeah, we do see more taxonomic similarities in terms of fishes when we're looking at shallow versus deep reefs, as opposed to deep reefs versus shallow cold water reefs, because the latter category usually has its own specialized assemblage of species that you don't find anywhere else, even in coral reefs.
Yeah, that's really interesting. And then that means you also have a very different set of functional roles being carried out, you know, temperate versus tropical. But how does that translate then to, say, on this reef, you would have, you know, a range of fish occupying different roles, playing different roles on the reef up shallow. Would you see those same roles reflected down here, or would you see, I mean, I guess it's sparser to begin with, so you'd see sort of less of everything being represented, but would there be some sort of mirroring of the same kinds of roles being played? You know, apart from maybe you don't, I guess you wouldn't have herbivory because you can't have photosynthesis down here, but you know, you might have more detritivory, or you might have more carnivorous species. What is it like? What are you seeing in terms of the functional makeup of this assemblage down here?
Yeah, definitely, because we're looking at a reef that now is kind of bordering, or, you know, outside of the photic zone. A lot of the primary production here stops, so you don't have herbivory. You don't have you have very little things that are planktivorous, so you don't have fishes such as, well, you actually, no, you do have planktivores here, but most of the fishes here are kind of adapted to more, I guess, predatory roles. They, you know, lie in wait for food to come by, like ambush predators. And then, you know, have things like epigonus, for example, they're pelagic and they feed on, you know, falling debris and other plankton from the surface. But yeah, by and large, you don't have any of those coral reef specific trophic guilds that you see up in the shallow realms, such as herbivory, and even coral eating species. They're kind of more absent here. I think even just generally looking at the reefs, you know, fish abundance is so low, and most of the fishes that we've seen are kind of sedentary, which I guess suggests quite a bit of, you know, the difference between a shallow water ecosystem and one that's in the Mesophotic or deeper.
Yeah, it's interesting. I was trying to, when I was looking, I was chatting earlier with Debs, and, you know, we're sort of panning around all this quite empty space. Even here, you know, I guess it's a wall, but you're seeing a lot of, you know, a lot less biological material, a lot less life. And so whatever's, all the processes that are going on are maybe slower and maybe more dispersed. Yeah, it's sort of interesting to try and understand, based on what we're seeing here in this glimpse of how this whole thing might work as an ecosystem. You know, obviously it's interesting geologically and geomorphologically as well. But in terms of it functioning as an ecosystem, it's just so different from what we're used to. We did see you mentioned planktivores and planktivory. We did see like a huge swarm of, I don't know if you saw the earlier footage of combination of marine snow and looked like crustaceans swimming around and worms and a lot of life in the actual water column earlier.
Yeah, I think I did see a little bit of that. And you do get planktivorous fishes down here, things that we've been seeing from, for example, like some of the MB8 and serenids that we've seen. They're kind of characterized by mostly plankton feeding species. But they also, are not obligate plankton feeders. I think a lot of them, like Plectrantheus, and that really mysterious purple antis that we've been seeing, they kind of look a bit more predatory. Like, they could probably eat something bigger, things that live in the benthos and crustaceans or decapods that live among the rocks. Whereas if you go up into the shallow water realm and you see things like Pseudanthias, other damselfish, general like Pomocentris and Chromis, they're strictly planktivorous, and you don't really see much of that down here in these sorts of habitats. That's actually a really interesting question because, if for people watching it on YouTube or Facebook, if you're a ecologist, who specializes in ecosystems and how things function, on an ecological level, feel free to chime in. I'm more of a taxonomist systematist, so this is a really unfamiliar field for me. I'm kind of just talking to you based on what I know about fishes and how they behave and what they eat. But if you have an ecological perspective on this, feel free to chime in and let us know in the comments, Facebook, YouTube. There's a really nice Plectrantheus on the topic of that, swimming into the screen now. It's been really difficult identifying species of Plectrantheus, mostly because there's known from so few specimens that when we see one alive on footage, it's really hard to tell if they represent new species or if they represent already described species that are just known from so little material. I can't really tell you if this is a new species or not, but it's definitely from the genus Plectrantheus.
Yeah, I'm a bit like you, Kai. I'm sort of an ecologist who works in shallow water on coral reefs, and I've done a lot of the surveys, especially since 2007. I've done a lot of the shallow water surveys on these reefs. In fact, we're probably not very far east from one of the cays or little coral reefs making up the big structure of Lihou Reef that we have surveyed actually multiple times. And it's difficult enough to try and understand the shallow water ecosystems out here in the Coral Sea because they're so isolated, and some of them seem quite unique. Some of them seem, surprisingly depauperate. Some of them seem surprisingly rich. And we're really only just starting to inventory what's where, let alone try to understand how it all works and how it's all connected. But having this another dimension by being able to see the depths now is great. I think it's just gonna be a great piece of the puzzle in our understanding of how the Coral Sea works, how the Coral Sea reefs work at least.
Yeah, definitely. This gets really evident when we start moving up into the shallower depths. For example, you see a really markedly different ecosystem coming into play when we're breaching the 200-meter mark, 250 meters even. You start getting things like Halimeda, some photosynthetic corals, and immediately, almost kind of abruptly, you see this big change in biodiversity. You start seeing way more fishes. You start seeing way more different types of fishes even. And that clear delineation between ecosystems can happen so quickly from 300 meters to 200 meters. That again, it just goes to show, there's just a very big play, or a very big difference in terms of what's going on in these kind of systems. And I think that's, to me, one of the more interesting and valuable things that we're seeing here with the Falkor dives, is that we see this really nice gradual change going from 900 meters all the way up to 100 meters. You're seeing this really cool gradual change in ecosystems and fish, coral, algae assemblages. And then you get a rapid burst of just diversity when you breach into the photic zone. And it's just, for me, that is just so cool.
So are you seeing some common patterns? I was lucky and unlucky because I got to go out to sea recently for 10 days with the Australian Institute of Marine Science, but it meant that I missed quite a few of these dives. Between, I think my last one that I was able to sit in on was Lihou Reef, and then what was after Lihou Reef? The one afterwards, but and then I missed a few, and so now I'm just back. So I'm just curious to know from your perspective, having been able to follow them, whether you're seeing really common patterns in what's at what depth on these reefs in particular, and whether that's different from what you might expect.
Generally, we do see, well, in terms of common trends, we do see a lot more biodiversity in the shallower realms, and that's to be expected. But what's most surprising for me is to see just the different types of fishes you see even at the same depth zone. So I think a lot of it has to do with habitat partitioning as well. Some of the species of fish, for example, like we've been seeing a bunch of undescribed damselfishes from the genus Chromis that Tiffany has previously recorded from her underwater bruff videos. They're very common at about 200 meters, but you don't see them everywhere. They kind of only appear in very specific habitats. And so I've been looking out for that kind of pattern. What kind of habitat dictates what kind of fishes occur in them. So in that regard, we're seeing some general trends, but we're also seeing some really interesting partitioning here. I think in the last dive, I can't remember where it was, we were looking at a mostly halimeda and over algae patch in the shallow realm, and we're seeing a lot of fishes that we haven't seen before in previous dives, where it was more characterized by hard coral and gorgonians. So again, super interesting, a lot of general patterns, but also a lot of patterns that don't follow, it's kind of specialized dive to dive, and that's pretty interesting. We got a comment here from Tiffany on YouTube. Hi, Tiffany. Thank you for joining us. Steeper is generally more diverse. There you go.
Yeah, I'm just thinking, I think we found that on the shallow reefs as well. You got some very exposed sides of those reefs, especially the smaller ones around the Herald Cays and Coringa, Chilcot, and the Magdalenes, and they were just these rolling plains of pavement, of carbonate pavement, covered in very low-lying things like just little foliaceous sponges and not much more, actually, little clumps of halimeda, and there was not much there in terms of fish life. And then as soon as you had the wall drop-offs, there it all was. And in fact, I was talking to Debs earlier, and there's a spot on the southern end of Lihou Reef where they still hope to send down a dive if the weather allows, where there seems to be sort of, a pressure point where things all come together and spawn, and everything's there, all the predators are there, and you do see that reflected down here too, right? So where you have these vast expanses of relatively flat or more horizontal terrain, and then suddenly you have like a block of something hard or a steep slope, and you suddenly get much more life of all kinds, not just the fish. So yeah, I think there's definitely parallels there in terms of habitat and niche partitioning.
yeah, the fishes do generally love ledges and things that are characterized by you know, a lot of substrate, a lot of complexity in terms of habitat. When you see a lot more just things in general living, I guess, in those kind of areas, as opposed to flat, monotonous rubble terrains or just, you know, sandy areas. So that's another interesting general trend that we're seeing, and I think translates, you know, not only for fishes but also in terms of corals and invertebrates. We do see a lot more life and diversity in areas that are characterized by more complex type of habitats.
We have a question online from Louise asking how shallow is the dive today supposed to go? And I think, I mean, usually you stop at about 2 to 100 meters, but maybe maybe the team on the ship could comment.
Yeah, the plan today is to go as high as we can. So usually that's about around 100 meters or just above. It really depends on the how hot the vehicle gets. So once we start getting above 300 meters of depth up into the shallow water, then the temperature of the seawater climbs, and then the temperature of the vehicle climbs. So, but yes, we're hoping to get up onto the top of the reef here. You can see our dive track plan for today is in that subset in Fledermaus, and so the vehicle's just gone all the way around this large rock, and we're going to start working our way up this S-curve. So this corner here where this slump has come completely down off of the reef, and so we're going to work our way along this wall to the south, and then start working our way up.
Thanks, Debs. Yeah, that should give us a nice range of different aspects as well. You know, that's the other thing we often found is that, you know, the minute you changed the aspect of where you're looking, you know, south versus north or even slightly southeast or whatever, things like that would suddenly create a whole different set of environmental drivers for things to then, you know, be affected by. So you then suddenly had a very different benthic community and then a very different fish community as well.
Hello, Falkor. This is Robin Beeman calling from Cairns. Who have we got online here?
Hey Rob, I'm here. It's Debs, and we have Daniela and Kai still here. So thanks for joining us.
Yeah, thank you too for to Daniela and Kai for coming on and helping talk through the audio and this vision. It's I've been actually being interviewed viewed for a TV show about this remote expedition. So I've been watching as it's been going along. I saw about half an hour ago just beautiful fringe of Brisington sea stars fringing over the rocks. That was a real standout, and still more Nautilus. But we've probably left the Nautilus now. What we've found is that in this, once we get up to around 500 meters, you're getting that big change of thermocline, and during the daytime, we're finding that Nautilus tend to stay below this zone. So I'd be very surprised if we see any more at this depth. So Kai, you're calling in from Sydney. What's jumped out at you at these depths over the last hour or so?
Hi, Rob. Yeah, calling in from Sydney. Again, we've been seeing quite a bit of that really interesting purple anti-iodine serenid. We're still not sure what that is. It could be a new genus. It could be something that's very poorly known. And I think for the first time today, we saw a juvenile of that fish, which I actually did take a screenshot and a screen video. I'm putting that up on Twitter now to see if my other colleagues have any ideas as to what that could be. We also had a really interesting ophidid earlier on in the dive that was identified by one of our watchers on YouTube at Nuni, I think her name is. So yeah, I think for me, it starts to get a little bit more exciting and familiar in the Mesophotic area. A lot of the fishes that we're seeing here now, admittedly, I'm not too familiar with, but it's still really great to see, people being excited about these kind of things. And me too, I'm really excited about all these fishes, coffin fishes especially. That's so cute.
Yeah, that's right. That coffin fish earlier, I'm not sure if people saw it. It just had the most brilliant colours. Probably the most colourful coffin fish we've seen yet in this whole expedition. I was really quite stunned by it, actually. They have been almost a monocolour usually, of course with those beautiful fringes and some really interesting appendages, but this one was utterly colourful. Quite a contrast to sort of the background white and grey and brown that we're seeing. And Daniela, what was as you've been on a few times this morning now? What's the highlight for you that you've seen?
Yeah, hi Rob. It's hard to say really, because I'm sort of spending my time looking at the screen and going, "Whoa, what's that?" Because I'm much more familiar with shallow coral reefs than anything that's happening down here. So it's been actually great to have a chance to chat with Kai and to hear what he has to say about the fish species and the fish assemblages down here. Earlier on, when we were at about 500 and something meters, and I was chatting with Debs, I was really surprised to see all the life in the water column, actually. There was probably a mix of marine snow and plankton, but it was just buzzing and dense. And then we also saw quite a lot of stuff on the sand, anemones and a couple of fish that seemed to be just hanging around on this really empty sandy plain, which is surprising because usually those are the areas where you'd expect to see less life. And I mean, for me, it's great seeing the Nautilus. I've sort of never really had a chance to see them like this before.
Yeah, I think we're becoming very spoilt with all the Nautilus. I have to think from biologically, from the whole expeditions, the Nautilus has really stood out for me. I mean, I knew that there was a population up at North Horn, but I never expected that nearly every dive we've seen them. But it's funny you talk about the sand. I actually really enjoy looking at sand when I'm scuba diving. Of course, the reefs get all the attention, but I find myself getting down into the lagoon and seeing what I can see down there. And you're right, it's it's not devoid of life. It's just it's quite different. So it tends to be more mobile. We've got lots of echinoderms, urchins and the like, sea stars roaming over the seafloor. In deeper depths, deeper than what we're looking at today, we tend to see a lot of sea cucumbers, and so these are the vacuum cleaners of the ocean, really. They and in places they've been quite thick, but because it tends to be a bit more stable down there, these deeper depths, the trails are often left behind. And in fact, I wrote a paper about these life trails, and they had this German name. It's called Lebensspuren, which is just German for life trails. And so there's this whole science around the marine life that you find crawling around on the seafloor. Of course, the animals have long since departed, but you can actually see the mounds, burrows, and tracks and trails that they've left behind. And so, there's a lot of thinking around who's left what behind. And I particularly like looking at the sand.
I think I remember that paper. I think we used it in, we cited it in that massive review that that we wrote. You were one of the authors, right? Rob was like, I think there were 43 authors on it, and it was everything we have ever learned about the Coral Sea. So anything that could be written about the Coral Sea is in that paper.
Yeah, that's right. But the big standout for me from that paper was just how little knowledge we knew about the deeper Coral Sea. So all of the focus was really around the shallow coral reefs and the ecosystem there. But once you got down below that scuba diving depth, it was virtually unknown. And so I think there were just one or two studies down at Osprey Reef, which I was involved with, and that was it. That was kind of the sum total of our deep Coral Sea knowledge. And yet with this latest Falkor expedition, it's completely change that around. It's a huge step change from the knowledge that we had before this year up to right now. I'm quite amazed at actually what we've discovered just in the last.
Debs, are you there?
Yeah, I'm here.
Yeah, so I think, was it Jim or one of the guys in the control panel, control room just now was asking what the difference between a coffin fish and a goosefish was. And I said that I think coffin fishes didn't have any lures. Actually, they do. They're really short and they're nested in between the eyes in a little groove. So they do have lures. As to what the differences are between both families from a taxonomic perspective, I'm not very sure because I'm not very familiar with either families. But yeah, they do both have lures.
Okay, great. Thanks, Kai. Jimbo is listening, so he'll have that update. Thank you.
Yeah, thanks. This is, I guess, the benefits of having lots of eyes from around the world and lots of questions. We really appreciate all those questions. But I'm no fish expert myself, Kai, but I'm just remembering that there was a large coffin fish. I'm pretty sure we saw on 364. Was that deeper canyon ROV dive that we did in the front of Diamond Islets across that very strange kind of chalky environment? It was around about 9 to 800 meters water depth. It's quite a large, pretty sure it was a coffin fish, and it had a very obvious lure sticking up from its head.
Yeah, I think I have that photo up on my computer at the moment, Rob. I think, have been seeing 2 different, or at least 3 different types of coffin fishes. I think the most common one that we've been seeing that really are the really big yellow ones. I think the one that you're talking about that you saw at the chalky kind of habitat, I think it's the orangey pink one that has blotches on the back part of its body. I'm not sure what the species is, but it looks different from the ones that we've been seeing today.
Yeah, it looks like I've got a big shrimp down here on the bottom left. Quite a decent size by the looks of things, at least 10 centimeters long. You're right. It is the same fish we're talking about. I remember we got a very, very close-up look at it. If you recall, we were literally in its face, but it was just quite amazing to see the lure structure. I was more familiar with like the anglerfish, the deep water anglerfishes. They have a similar lure, but this was just sitting there very, very quietly. Wasn't bothered by us with its lure. It would have been a good 5 centimeters sticking up from its head. Debs, you've got the 3D perspective up in the lower right. Could you just? It's not quite clear to me where the ROV is. Can you just, just move it around a little bit? Oh, okay, so we've just come off the back, have we, of the larger block?
We actually worked around it, so that we've done a transect sort of along the edge and come around the rock. And so we're just going to follow this debris field sort of up this direction towards the wall. And then work our way along the wall face here as it curves around before we climb up.
Okay, this is good. So this is one of the reasons why we chose this site, was because of this very obvious landslide scarp, and you can see it in the background. It's in a little bit of shadow, but you can see how vertically steep it is. This whole section of the reef, which is about 3 kilometres long, has collapsed, and we're just going over the boulder rubble remains. It's what we call a debris field. So what we're approaching now is what you might think of like a quarry face. So we hope to see, nice exposed strata. In some of the other ROV dives that we've done previously, when we've actually ran across the face of these, you can see the internal layering of the reef. It's almost like an X-ray of the reef, and so it's quite exciting to sort of go into this environment, and we're hoping to see similar layering effect in here. But one thing that strikes me though, Deb, so even though we have these great 3D maps from the multi-beam sonar on the Falkor, it sometimes just doesn't do justice for how rugged it really is at the seafloor. So we chose this ROV transect to go over these bumps, and in fact, from a 3D perspective, it looks like we're just going over a kind of a slightly rough seafloor, but in fact, the ROV is showing just big boulders and steep sides and cavities, and it's just jumbled mass of rocks. And so, like I say, it just the 3D map doesn't quite do justice for how rugged it really is at the seafloor level.
Yeah, I think some of that has to do with resolution, right? So you're in 300 meters of water and 400 meters of water, and we're up on the surface for that. So this grid that you're looking at is a 10-meter grid, so you're only going to see a boulder if it's bigger than 10 meters.
Yeah, it's still pretty good, of course. When you're talking about depths that are 0.5 kilometre deep, there's a limitation just with the sound acoustic sound beams, with the mapping systems, and so sound spreads as it gets further and further deep, and so your footprint of individual beams tend to spread as well. So there's a physics limitation to what you can achieve with sonar. when they're vessel mounted. It'd be different if the same multi-beam, a similar multi-beam was mounted on an autonomous underwater vehicle, for instance. And, you know, this is becoming more and more common in the world, where multi-beam sonar systems are actually mounted on underwater robots, literally flying just above the seafloor. And then you can get incredible resolution. You know, I've seen some pictures of the seafloor, you know, that are 3 kilometers deep, but you can resolve features that are the size of a briefcase, just because the actual multi-beam is transmitted from something that's just flying just above the seafloor.
Yeah, it's definitely amazing, the technology with autonomous underwater vehicles now, and even with ROVs they do it as well, map. So.
I'll just quickly jump in and say I've been asked on the YouTube chat about a link to that to our review about the Coral Sea with the millions of authors, and I've just sent a link via the live chat. But I was wondering if there would be a good way to maybe share the full PDF with people.
Yeah, I don't know. Actually, Daniela, if you send me that link in the Google chat, I can put it on YouTube. It blocks, we have filters on that, so people can't do a lot of linking to other things. So if you send me that link, I'll put it on YouTube and people should be able to see it.
Okay, great. I'll do that right now.
We'll have to redo that paper now, Danny, that we've got all this new data.
Absolutely. Yeah, it'll be great. I mean, we've also got a whole lot of new data from the shallow reefs. So I know that compared to these deeper areas, we have known a lot more about the shallow coral sea reefs, but really in comparison with anywhere else along the Australian coast or the Great Barrier Reef, we really knew very, very little until this last 3 years of these expeditions that James Cook University and the ARC Centre of Excellence for Coral Reef Studies have been doing for Parks Australia to basically to inventory and check what was on these reefs, because there's 30 odd reefs, shallow reefs, that are all at diveable depths. And only really a handful of them, and this review actually does a bit of an analysis of where most of the research had been done. And really only a handful of them had been surveyed in any kind of systematic ecological way, so that we could, you know, give them some kind of character and have some idea about how they might function or what state of ecosystem health they might be in. And so, yes, I agree with you, because we also know a whole lot more about the shallow reefs. And now this, I mean, having added this deep dimension is, it's just like, you know, a treasure trove of more information. So yeah, we should try and tie it all in and get it published.
Yeah, and I like the complementary nature that we've approached this this expedition at. So we reached out to Parks Australia fairly early in the piece, and we did ask that question: were there some reefs, the shallow reefs, this is, that had been studied and had stood out as sites that were, you know, worthy of visiting, you know, or particular parts of these large reefs, like for instance Lihou, it's 100 kilometers long. Where do you even start to look in the deep water? But we got some feedback really quickly, and it was quite useful. And this is the great link with you. For instance, they said at Moore Reef, one of those small little reefs that sit out to the northwest of the Diane Bank, they said that had really high, that was high in biodiversity in shallow water corals and fish. And as it turned out, it was you that had done the survey. It was great timing for us because we were in the vicinity and we did that first dive. You remember that up Moore Reef, this tiny little speck of a reef. It probably acts more like your classic seamount, you know, your pyramid-shaped seamount in the deep ocean. And certainly it was a really exciting dive.
Yeah, exactly. And in the shallows, it was also a really exciting dive from the point of view that, we're exploring something new, and we really, that I don't want to make assumptions, but I'm pretty sure that no surveys had been done before on Moor Reef. And we don't even know whether people have really dived there before. Maybe people going out spearfishing or yachting, but we don't know of any of this. So there was no documentation about anything to do with Moor Reef's life on the coral reef, shallow or deep. So there was this great parallel, right, between us getting to survey the shallow reef and then actually not that much later now being able to join you on the dive, on the deep dive up Moor Reef.
Yeah, that's right. I mean, the timing of all this is quite extraordinary. We think of COVID and the difficulty that presented with the Schmidt Ocean Institute, and we had this amazing opportunity to get the vessel out there. As it turned out, I was, I'd considered this as a potential proposal for the Australia's Marine National Facility, but knew full well it would take years for it to come about. And then, with this opportunity, we were able to very quickly pull together a proposal, and we were, very lucky that the Falkor was able to go out here. But I just think, what a resource it's produced. The 3D maps are extraordinary, and certainly from on shore, amongst the mapping people that I'm involved with, everyone is really quite amazed at the picture that's emerging of the deep plateau and the surrounding deeper flanks of these reefs.
Yeah, so those links to that paper should now be available, and there's other people also mentioning other papers that came out later. And yeah, if anyone has links to more coral sea papers, to write that review, it has a bit of history which I won't go into. It's boring, but to write that review, I ended up with a massive Excel spreadsheet of every single reference that I could lay my hands on, grey literature, published stuff, websites. And so I ended up with this amazing data set of anything that had ever been written about the coral sea. But it kind of ends in about, 2013, 2014, because I did have to do other things with my life. But yeah, I mean, I'd love to update it, because it's, now it is fairly dated now, and there has been a whole lot more research done since.
Yeah, and in my own field, I've mostly spent the time mapping the deep Great Barrier Reef, and so the Coral Sea was always just a little bit further out there, and no ships had ever been out there. But in 2017, I spent a month on the new German Sonne. It's a magnificent ship. It's one of the largest ocean-going research ships in the world. And we spent a month kind of hanging around in the Great Barrier Reef side, and spent a little bit of time just kind of skimming the edge of the western edge of the Queensland Plateau. So I was getting kind of hints there's something more there, but it was almost it's just too far to get to. And so it was always, gee, I'd love to go and explore this, but knowing full well that it would just take a huge effort to get there. Hence I just keep thinking how lucky we are to have partnered with the Schmidt Ocean Institute and the Falkor with this year-long series of expeditions around Australia. So we've certainly been the benefit, the beneficiaries of that, I guess, this unfortunate situation. But yeah, now everything has changed. We can understand these linkages. It'll help understand the linkages between the Great Barrier Reef and the Coral Sea. Of course, both the Coral Sea Marine Park and the Great Barrier Reef Marine Park join each other, right? They share the same waters. And in the deeper parts of the ocean, they share similar sediments and water masses. And so it really helps to kind of bring context for the deep Great Barrier Reef, because all of that water mass and currents sweeping in across the Queensland Plateau towards the Coral Sea. All the plankton that you see in the water column, it all ends up in the Great Barrier Reef as well.
Yeah, which is, you know, we were always curious about those linkages in the shallow ecosystems as well, in terms of the currents moving westward. And so, you know, thinking about, well, so are these coral sea reefs repositories for life, you know, on the Great Barrier Reef? Could they be, you know, as the Great Barrier Reef suffers these impacts, could these more isolated, more, you know, naturally protected from human impact coral sea reefs be refuges? But we're finding that there is like a coral sea assemblage, especially in the fish, that where there seems to be some kind of a break or a barrier, and you find certain species that just drop out in the coral sea. They're just not there. Whole families to some degree, and genera, and certainly species. Or you find a species that looks almost the same as something on the Great Barrier Reef, but is in fact a completely different species, just occupying that same niche. And only on the coral sea reefs closest to the shelf edge would you see one or two sort of GBR typical things, but then they drop out further east. And so there is both connectivity, but there also seems to be some kind of a barrier.
Yeah, I think we really need to get Tom Bridge on here and talking about this. I mean, Tom is the senior curator of corals at the Museum of Tropical Queensland in Townsville, and this is quite his thing. He's right into biogeography and trying to understand this connectivity, or I guess evolutionary variation between different sites. Oh, Kai, what have we got here? Is this a Ruffy of some kind?
Hi, Rob. Yes, that is a Ruffy. And that is all I can say about that fish, unfortunately.
Yeah, hi, gents. I was just going to say, could you zoom in? Thanks. So I like this theme of connectivity, Danny, that you've brought up. You know, some other things I can think of, for instance, you know, Richard, who was on here earlier, he's very interested in sharks, and he has a fairly large shark tagging program up off Osprey Reef and also on the Great Barrier Reef. And so he finds that there are the tiger sharks, which are the top of the I guess the shark food chain up here, that they're certainly the largest of the sharks. They roam from the Great Barrier Reef out into the Coral Sea. And so his satellite tags show them, for instance, feeding at Rain Island, which is the world's largest green turtle rookery, which is at the very far northern Great Barrier Reef. And during summertime, they can have, you know, thousands of turtles nesting there. And that brings the tiger sharks in. And those same tiger sharks, you know, once that turtle nesting season's over, then wander way out into the Coral Sea, you know, thousands of kilometres, and have been detected passing by these acoustic sensors that Richard's got set up around Osprey Reef. So I think that's fascinating that these very large animals are migrating to and from the Great Barrier Reef, you know, hundreds of kilometres in places.
Yeah, I think the same was found for, was it grey reef sharks from Osprey Reef? They were tracked, or even white tip reef sharks. Sharks, they're definitely considered much more sedentary, but it showed that at least up there, up around the latitude of Osprey Reef, there was a fair bit of this connectivity through the movements of large megafauna kind of things, like sharks and maybe even turtles, or, you know, the seabirds that nest on the Coral Sea Islands and that feed closer to the Great Barrier Reef. And so this sort of connectivity brought about by these larger animal movements. But then, you know, also the larval connectivity. Although, and I want to also touch back to what you said about in Tom Bridges' interest in the history of how these assemblages came about, because I think that's one aspect of the story that we're missing, because we don't know enough, right, about the historical development of these assemblages. But to some degree, I think there's a whole lot of random chance of you know, maybe one larvae makes it out to one of these reefs of a certain species, and it just so happens to be able to establish itself and form a population, and so then you have it there, but nowhere else. And I think that happens quite a lot on these isolated habitats.
Yeah, that's right. And we have this notion of, we've heard this before in other in seamount kind of studies with this stepping stone effect, where you've got these seamounts scattered across the wide oceans, but each one forms a little step from which to create, to gain habitat and life, and then that then allows the next step in the chain. And so seamounts have, you know, often thought of in this little oases of life through a whole series of steps. At each turn, you've got a mix of endemics, species that just never that are completely unique to that, and others that are completely cosmopolitan. You know, they're scattered right across the whole area. And you know, I guess we kind of go back to some of the stuff that Kai was talking about in other dives, where we've got home ranges of fish really broadly expanding from this expedition. So Kai, I think it might have been the previous dive, I was on there chatting with you, and suddenly you were talking about, I think it was like a little black damselfish, where you went a little bit crazy. You're talking about a home range that, in the Coral Sea, that had never been seen before. Do you want to talk about home ranges of fish, and how do you end up with a fish that is only known in, I don't know, thousands of kilometres away, suddenly on one of these reefs?
Hi, Rob. Yeah, I did go a little bit crazy about that blackfish. That was an angelfish from the genus Chilodontopus. You know, just like corals, fishes follow a really predictable biogeographical pattern, where you have species that are confined to certain regions of the world, and then they're replaced by sister species in adjacent biogeographical provinces. That could, you know, either be a result of historical vicariance or dispersal leading to speciation that follow these, you know, set patterns. What we're seeing with some of these mesophotic deeper water species is a much larger range, you know, for lack of a better word, expansion, where, you know, they occur in multiple biogeographical regions across the world. Why this is so, I really am not entirely sure. It seems as though like deeper water fishes have a much bigger geographical range than their shallow water counterparts. I did a bit of work during my honors on anti-tropical distributions. So that angelfish that we saw is potentially in anti-tropical taxa, which means that, you know, a species occurs on both sides of the hemisphere, but are not found within tropical latitudes. So we call that anti-equatorial or anti-tropical. That angelfish is known from southern Japan in the Ryukyu archipelago. It's also found in New Caledonia, and now, I guess, Coral Sea as well. Whether or not it's found in the regions in between these areas, we're not very sure. There's a very good chance that it does exist there, just that, you know, we haven't seen or sampled for it because it lives on such deep reefs. So yeah, it's a really cool biogeographical scenario here, where, you know, these could potentially be relictual populations of a once really widespread species that have now been separated and made disjunct by, you know, contemporary processes leading to these patterns that we see today.
Yeah, thanks, Kai. Really, it's great to get your insight on the fish distribution. If I put my geology hat on here, this whole Queensland Plateau is part of the Australian plate, which is moving northwards at about 9 centimetres a year. In fact, people probably don't realise that the Australian plate is one of the fastest moving plates on Earth. It's separated from Antarctica during the breakup of Gondwana in the late Cretaceous, and it has been moving northwards or sort of slightly east of north through all that time. And we believe it did its final separation from Antarctica about 40 million years ago. Back then, though, the water masses were quite cool. It was more at Australia was down at the sort of subpole climate zone, but it's only in the last 20 or so million years has the Australian plate, or in particular this part of the Australian plate, the Queensland Plateau, has actually shifted into the tropical zone. So it's the first time we got coral reefs growing, and we know this because there are some very, very large drowned reefs at the very up the northern part of Australia, close to Papua New Guinea. Ashmore Reef, Boot Reef, Portlock Reef, these are quite old reefs. They're probably sitting on a base that's around 20 million years ago. It's the very first time the waters were warm enough for shallow coral, tropical corals to grow. And so that's the sort of start time for this Queensland Plateau to even begin starting the growth of these coral reefs. But then over that intervening period, that 20 or so million years, we've had a series of subsidence events. Each subsidence event, the plateau itself gets a little bit deeper, but as long as the coral reefs themselves can stay growing within that photic zone, then they will continue to stay alive, and hence we're left with these 30 odd reefs that we have now. But yeah, so just to show that that land masses are moving up and down, but they're also moving quite, over a very long time, they're moving horizontally over, hundreds, if not thousands of kilometres. So, Debs, it looks like we're fairly close to that large scarp wall, if I've got that 3D perspective right. Is that the case? It would be just in front of us now.
Yeah, it's just in front of us. We're starting to walk, to fly over that way.
Okay, and the other thing people might notice is the sand between the rocks. Now, these are ripples. They're collectively known as bed forms, and they come in lots of different shapes, of course. But think of these just like mini dunes. And what it says to me straight away is that there's strong currents acting through here. So that's not surprising. It is essentially like a wall. This whole Lihou Reef is 100 kilometres long. It's in the top 20 largest atolls on Earth. So it's a very, very big coral reef system. And we're basically going up along the wall of it. So if you can imagine water masses sweeping out of the southwest Pacific, those currents, and it's called the South Equatorial Current, it hits this very large reef system and would wrap around it, and the currents likely sweep directly along the edge. And these bed forms are a real giveaway that we've got currents down here. So we're still moving through this rubble zone, and it can go a long way out. In fact, one of the real interesting things that we've been looking at during this whole mapping exercise is just to see how far out this rubble zone goes. And we've yet to really sit down and do all the measurements, but in some cases this rubble can be between 5 and 10 kilometres away from the edge of the reef. So that's a long way for debris to flow. Of course, it's just slightly gently downhill, but if there's sediment underneath it, there's no reason why big blocks can't slide for long distances. Can we just get a zoom in on these white corals, please? Okay, thanks. We're probably we're going to start. I think these are little stalastrid hydrocorals. My picture's a little bit grainy here, but this is about the zone that we start to find them in. And this is the lower Mesophotic zone. So we're passed through that thermocline, and temperatures will start to warm from here. And in this zone, we have been finding lots of these small, white, delicate hydrocorals. Stalastrids is their other common name. So, Debs, what's the temperature now at this 400 metre mark?
It's 12.1 degrees Celsius.
Yeah, we've definitely passed through that thermocline now. We're in the lower Mesophotic zone. It's just going to keep warming up. I think you said earlier that the sea surface temperature is around 25 degrees, but we'll start noticing big changes from here on. But yeah, these are stalastrid hydrocorals. We want to have a look under any nooks and crannies or any overhangs. We tend to find a lot more life in those overhangs, probably to do with the fact that this sediment is not accumulating there, and so we've noticed before in the other ROV dives, these crevasses and overhangs are great places to look for sessile marine life. There's quite an amazing big boulder. It's just sort of tipped over and it's leaning on everything else. So we're getting close to the base of this scarp. It just looks like, the giant's hand has completely scoured out the seafloor and dropped the boulders everywhere. But even amongst the boulders, I can actually see a little bit of strata occurring. Our strata being the layering. So these rocks limestone. Would have originally been shallow water corals or different types of carbonate sediment, now all compacted into rock. And you can actually see some of that what we call fabric poking through some of these boulders. Debs, you want to talk about your inset here, about the position of the
Sure. We just put up the this is the display that the pilot and co-pilot look at and keep a reference of where the vehicle and the ship are. So we're just, as you can see, headed for that lower left-hand corner is the wall, and the ship is to our left. east, and so we have that red circle that's sort of a keep out, hold off area. So we definitely always have to keep the ship and the vehicle in tandem as we move south along this track. So we're looking to get to the bottom of this reef wall slump feature, and then sort of follow it along as it curves around that slump structure, and then eventually make our way up the wall.
Yeah, thanks, Debs. And it's still windy out there, isn't it? How's the wind affecting you at the moment?
Yeah, it's actually pretty good right now. The weather's come down quite a bit, so not actually sure what the wind is at the moment. Can find that out, but looks quite nice and sunny outside from the camera.
And can you see the sand cay that's on the reef nearby?
Yes, we can actually. Let me see if I can get it up in the camera actually. Not actually sure if we can see it from where we are, but just from the camera view, we can if we go outside and look. But yeah, there is a bit of a sand key there.
Oh, this is nice. Can you just come in closer to that colony and have a close look at that? All right, so we might have some help from ashore here. I'm wondering if on the top these are harp ctenophores. Does anyone wonder if we can get confirmation from ashore or harp ctenophores? Have this distinct harp-like shape. They're quite odd looking.
Can we get closer or zoom?
They are really beautiful, aren't they? This is just beautiful to look at, like the translucence. We've seen a few of them earlier on previous dives, but haven't seen them clustered like this.
Rob, we got a ID from Louise on the YouTube chat. These are harp ctenophores. I think the species is Lihou Reef, based on Louise's comment on YouTube.
Yeah, fantastic. It's great to have this expert input here. First time I saw it, it was like I said, what the heck is that? But now we know. But really beautiful, aren't they?
For a while, the Steinhardt Aquarium at the California Academy of Sciences had them on display. And they come in all sorts of different colors: purple, yellow, pinks, reds. They're really beautiful.
Yeah, I might be looking at that same website. Most comb jellies are mid-water. So we've seen them, of course. They've got those lovely cilia that runs down the side. I've picked them up many times when I've been diving. But these ones have adapted to living on the sea bottom. But, you know, one by themselves, you can kind of make out the shape. But when they're clustered like this, you're almost led to think that there's some kind of sponge. But in fact, they're an entirely different kind of animal. Yeah, I see Louise is saying, yeah, they get them in the Philippines and they're very widely distributed. I've only ever seen them in the deep ocean here, so they're a real animal I'm unfamiliar with. But thanks very much, Louise. That's much appreciated.
So, Rob, personal question for you. What has been the one thing that you're just really dying to see on all these dives so far? Is there anything that you're particularly interested in looking out for?
Yeah, good question, actually. Of course, I always say the giant squid. It's like the giveaway. But, in fact, the cephalopods have been really great. And I'm not a biologist, but I have really enjoyed looking at the cephalopods, and they've often jumped out at really sort of odd places. Like even I think it was more reefs, Danny. We're almost at the top. We're just about to recover the ROV, and suddenly there was a big octopus that came out. So I thoroughly enjoyed seeing them. I don't expect them now. They just seem to pop out at really odd places. Geologically, I've been more than satisfied. For me, the striking thing is just how much material has been shed off from the edges of these. It's kind of hard to get, I guess, to get across to people just how amazing this 3D mapping is and just how complex the seafloor is. Like when you start with such a blank slate, like, a white canvas, so to speak, that's what we had before the Falkor arrived. We only ever knew, we only had the top shallow portions of the reefs mapped from airborne LiDAR bathymetry, and I'm quite familiar with that. But that's down to maybe 30, 40, 50 meters at the most. It's just this whole other world that's been exposed since then. And, every day we have this discussion with Debs, with the other principal investigators, because we're talking about forward planning and where an ROV site might be done, and we bring up this 3D view on our screens. I'm watching from home, and Debs is showing us from shore, and I think I just say wow all the time. It's like words are hard to describe the how amazing the seafloor is and the pictures that are emerging. So I can't say there's anything I'm really, really looking forward to so much, because literally every day is a new picture, a new surprise, or sort of reconfirming some of the trends that we've seen in other dives. But again, it's just adding all detail to what was really just this completely blank canvas before the Falkor arrived. Yeah, okay, Kai. So is it you saying you're looking for a coelacanth? All right, we haven't seen one of those yet. Let's keep an eye out for it.
Yeah, so I opened up the same question to our YouTube audience, asking the people watching in from home what they would love to see in the dive. And Louise said Latimeria, so coelacanth. That is something that I've been secretly hoping to see as well. I mean, given the rich biogeographical history shared between Australia, Indonesia, and the Solomon Islands, it is not impossible. But some other people here are saying they would love to see oarfishes, barrel eyes, a large ray shark. We also have another viewer who also wants to see a giant squid. As for me, in terms of what we've been seeing fish-wise, I've been pretty satisfied. We've seen some really good species, potentially new species, range extension records, even just fishes that are really rare that we don't see very often.
This looks like a gurnard to me there, Kai. Do you remember if it is, do you remember that gurnard that we came across? It was probably 2 dives ago. It was sitting on the seafloor and it's had its pectoral fins fully spread out. It was very, very pretty. I remember that.
Yes, we had a triglyph. So a gurnard. from a few dives ago, and they have really interesting modified pectoral fin rays that are detached from the main pectoral fin, which they use to crawl around the benthos that they live on. There is actually a really rare species of gurnard that has been making its rounds on Twitter now, called the jagged head gurnard, and it's also found in Australia on deep continental shelves. That is something that I'm also hoping to see in one of the dives.
Well, in terms of prettiness or sort of standout fish, I think that big eye that we saw on the last dive was a standout for me. If you recall, those beautiful pink and white stripes, a huge eye, of course, quite a large fish, and we got a very, very good look at it.
Rob, I'm just going to jump in with the temperature. Sorry, guys. Just going to jump in with the temperature here real quick. They're asking on YouTube, it's 15.4 degrees Celsius.
Thanks, Deb. Yeah, that big eye, Pristigenis mayeri. I will never forget because it was a very embarrassing moment for me. I was watching the live feed commenting, and because the feed that I was getting through Zoom was so fuzzy, I thought it was an octopus and only realized it was a big eye a few seconds later. And somebody on YouTube made a meme about that. And so, pretty funny.
Yeah, I remember that. So just for people ashore, you have to appreciate we're sitting at our desks, scattered around the country, Australia, and we're trying to talk to vision in real time. And so, it's not quite as sharp as what you might be seeing on the final stream on YouTube. So it can be a little fuzzy. And I've been caught before going, oh, what's that? It's something completely different. It makes you look a bit like an idiot, but if people understand, we're not getting a super crisp picture ourselves, and we just have to make do with what we've got. But yeah, I remember it was funny. Kind of blows your credibility, doesn't it, Kai? When you misidentify a big eye for an octopus. So,
mate, honestly,
anyway,
my credibility is out the door now with all these deep water fishes that I'm just wrongly identifying for everyone. So I'm just waiting to the Mesophotic realm with things that I'm familiar with to try to regain some credibility here. I also just saw a comment from Louise saying the water is a bit too warm for potential coelacanth habitat. That's quite that is quite sad to hear, because now it just completely ruins it for me.
Yeah, but we shouldn't lose hope for sharks. There have been some surprises. Even in deep, we've seen some deep water catch sharks, some other strange sharky-looking sharks, as I could like to call them. Richard Fitzpatrick, who is our other principal investigator, he runs a fairly large shark tagging research program in the Coral Sea, and so I always enjoy listening to his insight on the shark fauna that we've seen. Of course, remember the ROV is pointed mostly at the seafloor, and so there are these pelagic sharks that just kind of cruising past. That we've seen a few of these benthic sharks basically sitting down. Do you remember that one, Kai? It was quite mottled in colour. That was interesting. But yeah, we haven't seen any of the really big pelagic sharks yet, and it's possibly because of just the environment that we're focused on. We're looking at the seafloor as opposed to, sitting out in the water column.
Yeah, they're probably circling around the ROV. You just can't really see them. I am hoping to see a goblin shark, Mitsukurina austini. They're found in the Coral Sea as well, and in other parts of Australia. And they are deep dwelling species. So that has been on my radar as well.
Yeah, we know the cookie cutters get down here because there were some collected in the Sidaris voyages way back in the late 80s. They were particularly, I think, very unfortunately caught in some benthic trawl sleds. And then, they're preserved in a museum. But Richard talks about some baited traps that he put down at Osprey Reef, and they came back with all mangled up and with cookie cutter shark marks on them. So they definitely exist out in this environment. Perhaps just not here, against the sides of the reefs. Maybe they're more out in the plateau proper. It's about 1,000 meters deep, and they might be just roaming out in that more wider expanse. The other person who's done some work out here is Professor Justin Marshall from University of Queensland Brain Institute, and he was very interested in, fish vision and the like. But he had a really impressive set of landers, benthic landers, with special red lights on, and he was dropping them out at about 1,000 meters out in the Coral Sea. And there's some really interesting footage because these things, they just sit down there for a couple of hours. They've got some food and they just wait to see what comes along. And some very large 6-gilled sharks just came out of the gloom and started eating the food. And, to see them is, they look utterly primitive. They've got this big blunt snout. They're very, very large. They move quite slowly. Quite a different motion to what you're used to with these, the larger, the more pelagic, shallow water sharks. Boy, looking at this this rock wall now, it is completely vertical and very.
Hey Rob, I think we've lost your audio. Just so you know.
Testing 1, 2, 3, 3, 2, 1. Can you hear me?
I can hear you now. Thanks. Yeah, you just cut out in the middle of your sentence, so I just wanted to make sure there wasn't something wrong.
Okay, so Debs, we're right at the base of that big scarp. Is that right now?
Yeah, we're just working our way along it. Here, let me change the screen for you. Here, let's do this.
Yes, I see. We're just at the base of that cliff. And so what is your intention? You're going to just slowly traverse along the base of it, are you?
So Russ says that we're traversing while going diagonally up. So we're going to kind of traverse this bit and then work our way diagonally across to that other face and then work up. So potentially do something like that.
Yeah, thanks. It's like standing at the bottom of a skyscraper and looking up, isn't it?
Yeah. Russ, because it's better than looking down.
Yeah, well, I've done some dives out in the Coral Sea. I'm sure Danny, who's listening, has done a lot more than I have. But I remember swimming off the edge of Osprey Reef, which is really steep like this, and you get to a point around about 40 meters depth, which is it's starting to become a little unsafe at those depths, and you're just looking over the edge, and it's a strange feeling looking down into the depths as it sort of disappears and gets darker and darker. And I remember feeling like overwhelmingly drawn. I just felt like I needed to go deeper, but of course it's sort of like, no, I need to get up now. And swimming away was almost like someone was hanging on to me, pulling me back. But it was a very strange feeling again, it's like flying off the edge of a building and looking down like that. So this is a completely different perspective, looking up into that view. And this is like 300 meter high cliff. So I'm seeing a lot of sand rubble in between the rock. Be interesting to see what that is, but it's likely it's just shallow coral debris, right? So all of that sediment has fallen down from the shallow coral reef system that's 326 meters higher up, and it's probably likely got lots of algae, coralline algae mixed in amongst it as well. And this is a feature we've seen on all of these reefs, that the sand is a mix of coral and algae, even at these deeper. I see Jeremy Horowitz has joined us. Welcome aboard there, Jeremy. Jeremy is our resident black coral taxonomist. So Jeremy's involved with this next expedition with the Falkor, doing sampling out in the Coral Sea. So we'll then have permits to actually collect physical samples, and Jeremy's doing his PhD on black corals, and so the samples are important because he'll be able to do DNA and molecular analysis on them. And I was lucky enough to spend a month out in the Coral Sea with Jeremy last year at the Tasmantid Seamounts expedition, along with Joanne Whitaker from University of Tasmania. And we were looking at the Tasmanian Seamounts, the very far eastern side of the Coral Sea Marine Park. It's a seamount chain, a proper seamount chain made by extinct volcanoes. And they basically line a big long string of them down that in the Coral Sea. Some of them have shallow coral reefs on them, but it was quite interesting. We didn't have any cameras, but we did a lot of rock dredging, and Jeremy was on board as the sole biologist, and he was looking for these elusive black corals. But in the 56 rock dredges we did, I don't think we found a single black coral. So, but his input was invaluable nonetheless. So yes, Kai, we're getting into your zone now, aren't we? This is this lower Mesophotic. We're down at around, it's probably around 15, 16 degrees Celsius. We're going to get into, we'll be able to see the lower, the photic zone starting to come into view. Just get the first hints of coralline red algae. That tends to be the lowest, absolute lowest limit of light that down in these depths, probably around that 250 meter mark. But still, it's still dark. It's still quite the twilight zone. But you can see the soft corals, and there's more and more more and more soft corals appearing on the rock. So we're definitely heading into that lower Mesophotic zone now.
That's a really nice snapper from the genus Pristipomoides. That's Pristipomoides donatus. Definitely not an octopus.
Yeah, great, Kai. No, I think a bit of humility here is good. You know, we all have to acknowledge that we don't know everything, and so I just reach out and thank all of our experts ashore. The more eyes, the better in this field. We're all seeing this for the first time, and so we really appreciate all of our experts ashore helping us ID this really quite strange marine life in a lot of places. So, Kai, one of the things we're looking for on this particular part of the transect is to look for layering. It hasn't been really obvious to me yet. It could be that when you're that close to the rock, you're not going to see it. But what I'm seeing now is it almost looks like a conglomerate. Like this is classic, just large cobbles or boulders all cemented together, and that's quite possible. Remember, these is all reefs are sedimentary rock. Built up from layer upon layer of limestone and calcium carbonate. But as we come around the corner, I'm seeing more and more this strata appearing. Now, this is exactly what we've been looking for. You can see the layering there. In fact, this sort of harder and softer layers, and over time they erode, and you end up with this kind of almost wedding cake layer effect. So you kind of have to sort of stand back a little bit to see it. But in fact, what we're seeing is probably like a cut, an X-ray through the rock, and you can see those layers. I guess from your perspective, this is just another habitat, and there's a nice fish sitting there on top of the rock. But from a geological perspective, this is really, really interesting. It's angled down, though. It's not horizontal. So you can actually see from the top left down to the bottom right the different layers.
Yeah, and I think we had a question earlier on today while you were filming with Richard by Alison Bronson, who is a paleoichthyologist from the US. And she was asking if you had any idea how old these rocks are. This was in response to a question by someone on YouTube asking if it was possible to see fossils of large vertebrate mammals being preserved in the rock strata.
Yeah, good question. But no, there's no large vertebrate animals in here at all. This would have been fully marine and never connected to any of the mainland, like Australia, right? So these always formed out in the deep ocean, and in fact, there's no evidence of any humans out here on any of these sand cays. Not until, oh, look, there's a Nautilus shell lying on the seafloor. We've just passed over it. So this is quite interesting. Geologically, we've got the layering, the rock layering, which is what we'd hope to see on the right-hand side. And there's a Nautilus shell sitting on the seafloor. Now, Richard talked about this earlier. When they die, they can be gaseous, and they have air in them, and they just get washed ashore on beaches. In fact, I've got a couple sitting on my mantelpiece, and I just picked them up on Sandy Beach, up here in North Queensland. Here's one right in front of us, and you can see it's fairly eroded, like it's broken apart, but you can see the tiger striping sort of coloration on the side. It's definitely opened up. It's quite an old shell, but quite distinctively a chambered Nautilus. But as far as the age goes, yeah, like I say, these are always formed out in the ocean. There's been no land animals, fossils on them. But in fact, the sediment itself would be filled with fossils, but it'd be coral. It'd be shallow water corals and algae and halimeda, which is a green algae. In fact, we can see these white flakes on the seafloor. It's very likely halimeda algae that's come down from above. And so if you in fact, Jody Webster, who is a principal investigator with this expedition, this is his game. He's into fossil coral reefs, and he's made a career out of sampling ancient fossil reefs to see what kind of species have made them up. And so you can do this. You can take a core or a rock sample, and you can actually see the original coral colony within. They can work out genus. They can even work out roughly whether it's a shallow water coral or something a little bit deeper in the water column when it was living. But now when they die, of course, they get locked up into these layers and layers of sediment that build up through millions of years. So the rock that we're looking at here is perhaps millions of years old. We know that the top parts of these coral reefs are probably Holocene in age, maybe only grown in the last 10,000 years or so, but they're growing on top of a much, much older reef edifice. These large atolls and banks, millions of years old, and that could be the age that we're looking at here.
Yeah, that's very cool. Look, I'm not a geologist. I know very little about rocks, but the layering and the stratification is very clear even for someone like me, and it really does look like a wedding cake.
Yeah, I'm glad you can see it too. I have to kind of stand back a bit and think about it, but I've had the benefit of doing this for many, many years. The other thing I'm enjoying is the marine biology side of things. It's the little habitats, those little micro habitats, those layers provide just a home for a whole range of animals. Like we've seen the fish just using the ledges for resting. Under the overhangs, we see lots of corals being attached. So I guess the complexity of the different layers helps to create more of that localized habitat, which attracts more and more marine life. So I've enjoyed watching, I guess the connections, the relationships between the marine life and the rocks themselves. So just talking about this, I guess this notion that all these reefs are built up upon layers of coral. I remember I can't remember which ROV dive it was exactly, but as we were flying up this steep wall, very similar to this, a big coral colony, very obvious. It's probably a favid, because I think Daniela was on at the time, and she said, "Oh, that's probably she could recognize it." So this is a fossil coral colony embedded in the rock, but it was still so well preserved that you could make out the genus of it. So that's, that was a really obvious example.
Yeah, I remember that dive, Rob. It was 800 meters, if I'm not wrong. I think it was Platygyra or some other similar-looking coral from the same family.
Now this looks a bit like an acropora, but if it is, it'll just be dead and probably just fallen down from above. So you can see this white sprinkling, almost like snow on the rocks. This is sediment that's just come down from above, but it would likely have, shallow coral bits as well, including larger colonies. People also have to remember that this is the cyclone zone, the cyclone belt, hurricanes, and every year we get cyclones in this area, and we've had some very, very big ones sweep through here in the last 10 years or so, like Cyclone Yasi. I remember it was like a category 4 or 5, and they've swept right over these reefs. And we've been back and visited these reefs, and you can see very large chunks of rock have been pushed, literally gouged out of the top, just because the wave energy is so high, and it's actually chewing out tops of the reef, and so you can end up with a lot of material being mobilized after a cyclone event. So it's not surprising that even here, we're seeing this sediment kind of raining down from above, but it could be just normal wave action, or it could be from the latest cyclone. Debs, or whoever's in the ops room, could you just show us the 3D map of where the ROV is at the moment, please? Thanks. We're still walking around the base of that skyscraper by the looks of things. So that's a nearly 300-meter high cliff, and we're just still at the base. Yeah, and we're getting lovely collections of soft corals as well appearing. And I'm guessing maybe one of those was a black coral. If Jeremy Horowitz is watching, I wonder if you could just confirm if one of those was a black coral. Yeah, so for people watching ashore, if you have a look at that inset, you can see we're going around this, we're about to go around a bit of a corner, but we're still at the base of the cliff. And the aim is to sort of work our way slowly up to the base of it. I think it's probably around my guess is probably around that 200, 250 metre mark. And then there's a very steep rise to the top. We've very rarely gone any shallower than 80 metres, and that's usually because there's shallow coral reefs quite close, and there's a safety factor here. It's also quite windy up there, and so there's a lot of operational considerations about where to place an ROV. We're not going to get right up into the real shallow coral reef system. We just can't do that with this fairly large ROV. The other thing is the ROV itself starts getting hot inside, and so we just can't linger too long in those really shallow waters because it's so warm. Okay, looks like we are now starting to climb that wall. So it's very, very steep. It looks almost vertical, both in the 3D perspective and also in the ROV vision. And so we'll see the temperatures climb and a high rate of depth change through here. Okay, thanks very much, Deb. Starting to see those sea whips occurring. Down in the cold water zone, these would typically be bamboo coral, but up here now we're getting into a soft coral world, and so these sea whips start becoming more prominent, but they're very different to the bamboo corals we find in the deeper, cooler waters. Can we just have a closer look at this, what's growing on the rock here? Thank you. Paul, could you just take off the inset, please? Yeah, thanks very much. Appreciate that. I like this. Crinoid condo, absolutely. In fact, all these colonies just covered in commatulate crinoids, using the colonies to push further out in the water column to catch their food. Jens, I'm wondering, are we seeing currents increasing now? I'm just getting the hint that some of these colonies are starting to wave a little bit. I'd be curious, are you experiencing current down at this depth?
So, I didn't have the microphone turned on. Okay, so Russ, our ROV pilot, says he's experiencing currents, and you can see the effect of them on the umbilical coming down to the top of the vehicle. We've got a camera looking at that. Our ADCPs aren't doing so well today, and we're only getting currents information down to around 150, 200 meters. So we can just do it by eye from the vehicle, unfortunately.
Yeah, okay, Paul. No, I just it's good to know this. Of course, this affects, I guess, the heating the internal heating of the ROV. It's quite a juggle between depth and wind at the surface and keeping the ship safe, and because the overall heating internally, and because it warms up as it gets, it has to work harder and in higher currents. So I'm trying to make out what we're seeing on the seafloor here. Most of this white material would be shallow water coral or algae that's just fallen down from above. That's your classic kind of white limestone. It'd be fairly fresh too, I would think. So this is, there's quite a lot of, just about every surface is draped with this white sediment. So, as contrasting the darker, older rock underneath.
Butterfly fish from the genus Roa. This looks like Roa excelsa, which is a new record for Australia, but there has been some contention going on with what this fish could be. Previously, I've identified this as Roa australis, but based on some of the 4K videos that the control room people have been sending over, this doesn't look like that species. Yeah, so I'll just be conservative here with this one and just say Roa butterfly.
So, Kai, is it normally seen at this depth?
Yeah, roller butterflies are deep water species. One in Japan gets to shallower depths, but it's because the water temperature there is much colder. But in general, roller butterflies are mesophotic and deeper.
Okay, thanks. Well, the temperature's steadily climbing. We currently got a water temperature at the ROV of 17.8 degrees centigrade.
Yeah, well, I guess that's comfortable swimming. It might be a little bit chilly, but it's definitely warming up. At the sea surface, I think said earlier about 25.7 degrees. So it's very warm and be quite comfortable swimming at on the ocean at the moment.
So on the YouTube chat, we're joined by Brian Green, who is possibly the best mesophotic coral reef diver I know of personally. Brian has collected hundreds of undescribed species from all over the Pacific, diving down to depths like this on Rebreather. Louise and I named the species of fairy wrasse after Brian this year, Simulabris Brian Greeny Eye. So welcome aboard, Brian.
Yeah, yes, welcome aboard, Brian. It's interesting that you've got this such a connection, also having a species named after you. I'm really enjoying having the fish army, as I call them, join us because when we started this whole expedition over a month ago, we didn't have any fish biologists on board. In fact, we had very few ecologists until we had Daniela Ceccarelli join us and to join Tom Bridge and Merrick Eakins from the Queensland Museum. So I've quite enjoyed seeing the biology team expand, and a big part of that is the fish army ashore. Learning about all these new fish species and range extensions has been a real thrill. So I'm not noticing much strata here. Imagine we're actually flying along the front face of this very large scarp feature. It was earlier in the dive, we did see the layering effect. Here it seems to be a bit more massive. But something that is standing out is all this pitting that's happening in the rocks. Now, that could be a result of when it was subaerially exposure. The subaerial just means exposed to air. Now, and it's quite possible that in the long geological past, and I'm talking over deep geological time here, that that this whole rock surface may have been exposed, and so we're getting kind of that limestone karst-like effect. But there's definitely more pitting appearing. These are little holes, these tiny little nooks and crannies that are now appearing on the rock itself. And you can see from the inset how steep it is. So we're not even halfway up that steep wall, and it is vertical in a 3D perspective, and clearly it's vertical in the ROV perspective as well. And so now we're going to start rising quite quickly, working our way up the wall into the shallow water environment. The other thing I'm noticing, Kai, is this sort of reddish tinge on the rock. Now, this would be coralline algae, lithothamnion likely, and it just shows the very lower limit of light it's penetrating there. If the lights were turned off, it would still appear quite dark, but there may be just the very, very hint of sunlight at these depths. I'm sure deep Brian Green, who's spent time down here on mixed gases and rebreathers, would probably be able to more experienced than most people in these depths. But there is enough light for a bit of photosynthesis, but it's generally being done by red algae, and there's these red patches that we're starting to see on the rocks. There we go. That's with our light out, and you can see the laser beams in the.
There we go, so you can just about make a few things out with the light from the surface now. Having to open up the iris on the camera quite a bit, but we are getting stuff. Ah, yeah, I'll change cameras over. Hang on. There we go, there's a situational camera there. Slightly different view.
Yeah, thanks, Paul. And this is without lights, obviously.
Yep, all the lights are off right now. And in fact, on our rearward-looking camera, we can see light coming down from the surface. Let us know when you want the lights back on.
Yeah, okay. It is a very different perspective, isn't it, when you turn the lights off? Yeah. Okay, let's bring the lights back on and start looking for fish. But really, it's very interesting. And of course, this is just the very lowest limits of
lights, guys.
Light filtering to the seafloor. But of course, the marine life are now able to make use of this for photosynthesis. And this is this red algae that I mentioned earlier. And you just see the tinges of it appearing. So just behind that big that coral colony that we passed over, that rock looks like just a big jumbled mass of shallow water coral colony. It's just tumble down as a debris chute, all piled up on each other, and it contrasts quite well against this background darker rock. Thanks, gents. Yeah, if you could just do a sort of slow zoom over this colony and its crinoid inhabitants as well, it'd be very nice to have a closer look. I think we said a crinoid condo earlier. Yeah, thanks, gents. Really beautiful. I'm trying to get Tom Bridge to come and join us. Tom is Senior Curator of Corals at the Museum of Tropical Queensland. This is certainly his zone, the Mesophotic zone. Let's see if we can follow, chase him up.
Yes, Luis, it's 21.1 degrees centigrade.
Yeah, it's definitely board shorts water. You could easily swim in these conditions now. But summertime here off North Queensland, it can get up to 30 degrees. And so it can be very, very warm. Mentioned earlier, when we go scuba diving, it's just usually a very thin wetsuit, maybe 2 or 3 mil at the most. And it's not really so much to keep us warm. It's just to keep us from being sunburnt.
Could we have a closer look at those fishes, please, that just swam by? They might be gone now, actually. And yeah, Brian's comment on YouTube was actually quite literal. You should see some of the videos of him and Richard Pyle diving in Mesophotic waters. Oh, can we get a zoom on that Rass, please? To the right. I think that's a Caelinus.
Yeah, there was
Can't really
another.
know. Yep, sorry, Rob.
I was just saying, gents, it was, if you can pan right, that other fish, the Kaisaf, went into that big boulder on the right-hand side. It may have gone now.
Yeah, it's a bit fuzzy on the Zoom. It looked like a Caelinus, but I'm just waiting for it to pop up on the YouTube live feed to confirm.
Yeah, thanks. Could you just zoom in on this this object? I can't quite make out what it is. It might just be a rock with a algae on top of it. Oh, never mind. We've passed over it.
Yeah, so both of those Rasses just now were from the genus Oxycaelinus.
All right, the inset view shows we're about halfway up that wall, but it looks like the steepest bit is right in front of us. So around just, oh yeah, it's perhaps a little bit, it's not quite up to date, but so we're seeing 166 meters, but we've got this very steep vertical wall right in front of us, and that will take us up into the shallowest part of this dive. Okay, thanks, Paul. If you could just remove the inset, thanks. You can see how active this feather star is. It's taking its arms and folding them in because they're completely mobile. You can see the little cirri actually hanging onto the coral colony here. In fact, they can swim quite well. If that was bumped and was dislodged, it could actually use its arms at this beautiful waving motion. It's quite beautiful to watch. I haven't seen them do this so much out in the Coral Sea, but an expedition I was on in Antarctica, where there were lots of commatulate crinoids in that very, very cold water environment. Whenever the light from the ROV got close to them, they would all jump up into the water column and start swimming in this beautiful, graceful motion away from the camera. Can you notice how green suddenly the rocks has become? It's all green algae, obviously, but it really jumped up in the last 40 meters or so.
Yeah, someone on the YouTube chat has pointed this out as well, that the rock's getting greener and also purpler, more purple. Yeah. There's a little fish there in the hole. Can we have a zoom on it, please? Oh, never mind.
Is that the fish you're after, Kai?
Can't really see from the live feed.
Yeah, orange, and we're getting a lot of thermal effects in the water.
There's a
Orange.
thermal climb here.
Yeah, possibly.
Oh, that's okay. Yeah, I see it. That's Plectrantheus bennetii.
Right, is that the one you're after or not?
It's a different one, but it's a good fish either way. The other one was a bit lower in a cave.
Okay.
A fish is a fish. I'll take it.
Okay, fair enough then. Just shout out if you need us to stop. Yeah, the temperature's gone up from 22.2 to 24.4 now.
So, Kai, you're looking for fish. I'm looking for something called halimeda, which is coralline green algae, and we've seen them growing down to around about 100 meters. So it's still a little bit deep for them. But I would say that probably a lot of this white sand has actually come from living halimeda. That's when it dies, it breaks up in these little cornflake-like layers, little flakes about the size of your thumbnail, and can make up a very, very large proportion of the sediment coming off these reefs. In other dives, we've seen them like curtains hanging down from around ledges and the like. They'll be very obvious when we see them. So I'm looking kind of for the lower depth limit of when we start seeing them living.
Yeah, I like halimeda too, Rob. They make really good habitat for certain species of fishes. My study group, which is what I'm working on mostly for my PhD, Rasses from the genus Cerulabris, and they're very fond of halimeda beds and exposed rubble outcrops where halimeda likes to grow in. So I'm looking forward to seeing more halimeda too.
Yeah, on our previous dive, that was 365. If you recall, we were up in that on the very front face of Diamond Islets. This is a few days ago now. We went through that very sort of gentle gradient up into that 100-meter zone, and we went through a distinct halimeda zone. It went from soft coral, then rhodoliths, and then we're into that Leptocerus, which is a scleractinian, a zooxanthellate hard coral, very plate-like. And then we went into that halimeda meadow behind it. So there were some people watching, Marty McNeil in particular, a world expert on halimeda. She got very excited. It was quite amazing to see a big meadow like that, just sort of sitting on the edge of these big coral reef atolls. But we're seeing it in the sediment, but to actually see the living meadow was something else. And I guess the big thing there was the gradient was so gentle that we were seeing those zones stretched out. And it was the first time we'd really noticed that on all these ROV. There's a lot of colour coming in now, isn't there, Kai? It's really obvious.
It's almost like a painting. It's like a Picasso painting.
Yeah, but I guess...
Yeah, go ahead.
Sorry, go on.
No, you go.
I was just saying we probably need a little bit more habitat change, a bit more structure, maybe nooks and crannies for fish. Would you agree? Haven't seen a huge number of fish on this particular dive.
Yeah, there's not a lot of complexity here, which is why we're not seeing a lot of fishes. I did see a bunch of teeny tiny ones. Impossible to approach though. We have a question from Tiffany on YouTube asking if the ROV has any blue light filters.
I don't think so. I'll just check with the pilots. Do we have any blue light filters on the cameras? No, it's just no filters all the cameras.
Hello, Falkor team, Rob and Kai. This is Jody Webster here.
Hi, Jody. Welcome to the chat.
Hi, Jody. Enjoying your rainy and cold Sunday afternoon?
Yes, it's rather miserable. I think I'd rather be in the beautiful tropics.
Hey, Jody, it's Rob here from Cairns. Nice for you to join us in this last stretch. We've had quite a journey. Remember, if you see any fish, just go quiet to allow Kai to jump in. They could pop out at any time. But yeah, we've seen some really interesting geology all the way up, which was the whole aim, to sort of capture see if we could see that internal structure. And we definitely saw that at the base of this big cliff.
So what depth was that, Rob?
It was probably around 300 meters. It was just as we passed onto the more flat face. So it was probably more that section of the scarp that was at 90 degrees to the reef itself. That was where it became obvious, and you could really see the layers.
Oh wow, very exciting! And they seemed to. I had a brief look earlier, and they seemed to be inclined, sort of dipping towards the slope. Is that correct?
Yeah, for sure. Yeah, no, there's definitely a dipping effect in there. So we need to keep an eye out for fish. There's a few down there.
They're very skittish and they're very flighty because it's not much going on here in terms of habitat complexity for them to hide in. So approaching fishes in this kind of area is proving to be very challenging. That's a male Genicanthus bellis. It's a swallowtail angelfish. The females should be around, and they're very different looking. The females look like a completely different species. They're black and blue instead of the orange and gray males.
Yeah, I did see another one before. Well, they're making use of these tiny little grottos, these tiny, small little cavities in the rock.
It's also really hard identifying them because this rock face is so vertical that a lot of the fishes we're seeing are from a dorsal perspective, which makes identifying them extra tricky through the live Zoom feed.
I think you're doing pretty good, Kai. You know, if you can identify male species just by a quick glance, that's pretty impressive.
I'm under heavy scrutiny from my compatriots on YouTube now, so I can't afford this stuff up.
Hey, Jody, we're heading into your zone. This is really interesting, about 120 meters. Do you want to tell people about the low last glacial maximum and low stands?
Yes, Rob. So we're actually about 123 meters in the Great Barrier Reef, where we've done work both mapping, but more importantly, seabed drilling with the International Ocean Discovery Program. We're able to recover cores, reef cores, through some of these submerged terraces, obviously not these vertical faces, but the tops of these sorts of structures. And we're able to determine the lowest extent of that low sea level position at about 118, 120 meters. And at the various dives out here in the Coral Sea, we have seen some interesting ledges and overhangs about this depth. So let's keep an eye out and see what we can see.
Yeah, that's right. Because it's so vertical here, I would think if there's any kind of ledges, it should be obvious. And I think that last dive, once I'll just be quiet for this.
Damselfish. It's not very. Yeah, this one's a bit hard to identify. The other one behind with the black tail, that's another Genicanthus species. That's a female Genicanthus melanospylos. This damselfish is a bit hard to identify, though. There's not many distinguishing characteristics for me to see. Brian Green suggests that the damsel is Chromis degroyi. So we'll pen that down as a tentative ID for now. Thanks, Brian.
Okay, Jody, for you and I, we're looking for sort of hints of any overhangs, wave cut, terraces and the like to demonstrate low stand sea level. So, 20,000 years ago, the last ice age, sea levels globally had dropped around that 120 meters. So we're kind of in there at the moment. You just see the hint of some sort of perhaps in cutting right there. Is it? What do you think there, Jody?
Yeah, there's some sort of indentation. I guess you guys have seen deeper, at least what I saw, it seemed to be quite a bit smoother and more massive deeper down. So perhaps this is some sort of indication of that. If indeed, this steep wall is indeed it's part of a landslide scarp, and that landslide scarp is quite a bit older. And if sea level then dropped to this position, this is obviously, oh wow, that's quite spectacular. This steep face from a substrate perspective is obviously not a great place for any kind of significant reef development. So wow, there seems to be some sort of structure there.
Yeah, that's right. A really obvious ledge, isn't it? So if you imagine the sea was lapping at the very, very base of this, like 20,000 years ago, that everything that was exposed would be slightly more eroded. It does, the face has this kind of pitted look about it, as if it was, once exposed.
Yeah, it's a great view of that wall.
Yeah, we first saw that big terrace sort of feature around that 120 metre mark up at Osprey Reef. And we actually had studied it in some previous expeditions. What was interesting about it biologically was the wave cut, this sort of low overhangs. It's almost like a rim of a bathtub, right? So you think of these big, big atolls, they stretch for tens of kilometres and you have these big vertical faces. And then there were these ledges around that 120 metre mark. And we saw this at Osprey Reef. And when we actually had the ROV looking in there were lots of stylastrid hydrocorals and lots of small corals sort of sitting up on the ceiling of those overhangs. So it was quite an interesting little sort of mini habitat.
Yeah, and this geometry of these atolls, or the flanks of these things, particularly the scarp, is quite different to what we've imaged and worked on in the Great Barrier Reef. I'm thinking the Noggin Pass area off Cairns and also the Hydrographer Passage area off Lakai. In there, the slope and the substrate is much gentler, and so as a result, quite significant fringing reefs during these low sea level times were able to develop. But this is probably not the case in this sort of margin and geometry, just because of the steep nature of it. Any kind of community, that was here would be very, very compressed and very, very, compressed and small habitat for fossil reef growth.
Yeah, it's a good point about this compression. On a vertical face, those zones are very close together. And the best example of that was, in fact, the last dive, 365. It was a very different kind of ROV dive. It was on a gentle gradient. And if you recall, as we got closer up into the shallower coral reef there, the zones became more stretched out and far more obvious. So it was quite interesting, the contrast between that gentle gradient and the steep wall.
Yeah, it would be fascinating to be able to, hopefully on Brendan Brooks' cruise in a month or so, try and be able to sample some of these exposed outcrops and limestones. And we could actually get some idea of the age and the nature of the deposits.
That is a Odontanthes. Can't really tell from this angle as to what species that might be. And the other stripy fish looks like a Rass. I'll wait for the live video. Sorry, I'll wait for the video on YouTube to make a better identification.
Yeah, Kai, if you see a fish, just sing out. If Jody and I are talking, we'll shut up straight away and you can take over.
Yeah, no worries. So I just had a look at the YouTube video, and the Anthias is Odontanthes bourbonius, which has a massive distribution. It's found all over the Western Pacific and even Indian Ocean to Africa. And the stripy fish looks like a Pteriolabris. Yeah, here's another one.
Just to let you know, they're trying to avoid moving the ROV too fast to try and keep the motors a little bit cooler. Things are heating out quite a lot here. The outside temperature in the water now is 25.6 degrees centigrade, and the oil temperature of the motors is over 50 degrees centigrade right now.
Yeah, thanks, Paul. Could you just linger around this really large colony here and see what we can see attached to it? In fact, what's interesting is the optics now. We're getting, I guess, density changes where perhaps less dense fresh water is mixing with warmer water, and so you get this kind of rippling effect. And this is just simple optics of, vision looking through one water density to another.
Oh, Christopher Ma would love to see this one.
As well as the big sea star, there is a fish knocking around. It was near the base of the coral. That's the sort of pinkish coral that's hanging down. I can't see it right now, but there was a fish as well. Just gonna try. Oh, there's the fish. It's down at the bottom by the yellowish coral. It was right about there. Yeah, I know. Oh, there's another one there as well. Russ has just found a fish in a crack. I don't know if you can quite see it.
Yeah, that's another Jenny Canthers angelfish, a female Jenny Canthers melanospylos.
And there's another one to the right by that orange coral. I don't know if you can see that on your link, Kai. It's just hiding.
Yeah, I do.
Behind, yeah.
It's a bit far out, and it's being blocked by the coral.
Yeah, it's just gone to hide. We'll keep looking for you.
Cheers. Yeah, this habitat's making it really difficult to look for fishes.
Can you just pan left, guys? There's a very large sea star here on the underside. It's quite beautiful.
I'm not very good with sea stars, but I think this might be Astrosarchus. And I think Brian Green also brought that up on the YouTube chat. Christopher Ma would love to see this. So if anybody's watching, could you please get a screenshot for him and put it on Twitter, please? Thank you.
Yeah, these overhangs are quite important, as we've seen in other dives in the Coral Sea, where we have these really large good-going fans hanging down. Probably the most obvious is in Osprey Reef, where you have these big overhangs and really large fan corals, meters across in places, hanging down.
Just a quick one. If you do want the 4K frame grabs, we can email those later on. Just drop us a message, Kai, and with roughly what times you're interested in, and we'll upload some 4Ks for you.
Yeah, can we just linger at this large colony that we're just passing over now? We're just about to stop here. This is quite remarkable size. Yeah, so request from Jeremy Horowitz. I think Jeremy would like to see if it's a black. Thanks, Russell. Any chance of doing a real zoom in on the thick branches? Jeremy can might be able to identify things just from the close.
Is that the one you're interested in, or the one on the left as well?
I'm pretty sure it's this one that Jeremy was interested in.
Okay.
I'm just watching the YouTube chat.
Oh, he's on there, right, just put yes, okay. It's a fun name on the on YouTube, the Pumpkin Star. I like that.
Yeah, it definitely had a pumpkin shape about it. It's huge, really. I mean, probably between 10, 20 centimeters across. Okay, so Jeremy says it's below this one, so we might be looking at the wrong colony here.
Hopefully it's this one. The one right we're zooming on now. There it is. Okay, so he seems happy with that. Okay.
Sure.
Well, that's the right coral. Let's just see if we can zoom in. Is any more zoom available, or? Yeah, let's get right in on there.
Thanks, and I'm also enjoying seeing the halimeda at the background. If you look through behind the coral colony, you can actually see the living halimeda. It's these little green flakes. They're all joined together. And when they die, the coralline algae makes up a big portion of the sediment on the seafloor. So we are definitely in that living halimeda zone. So 120 metres. That's good to know. If you look at the background, the whiter flakes are the dead pieces of the halimeda algae.
Do you think he's happy now, Rob? Oh, he says just checking.
Yeah, thanks. We'll keep going. Yeah, I think this is the exciting thing about all this. You guys are, well, for me, you're 600 kilometres away, and yet we can be sitting here and helping guide and ID things on the seafloor from different parts of Australia. So it's a really remarkable operation.
And I'm right in thinking there's never been another ROV that's dived down here. This is all the first, isn't it?
Yeah, that I'm aware of. Prior to the Falkor coming out here, there'd been one deep RV expedition done, run by the Germans, the German Deep Down Under expedition in 2009. That was just to Bougainville and Osprey Reef. And there had been some shallow ROV dives done to around 150 metres. And I was part of that with Tom Bridge. That was out to Holmes and Flora Reef, which is due east of Cairns. Other than that, I'm not aware of any other deep expeditions, and certainly no ROV work that I know of has been down here at Lihou.
So we're all seeing this for the very first time. Awesome. Oh, fish. Middle of the screen now.
Those are Klein's butterfly fishes, Chaetodon kleini. So this is not a mesophotic species. You find them in very shallow coral reefs as well, but they do get down to 150 meters, I think.
Okie dokie. We're just going to continue going slowly upwards. I think we can see the halimeda falling down.
Oh yeah, Rob, do we have a sense of where the top of the bathymetry stops here and meets the LiDAR? Are we able to see the depth at which the landslide scarp fails from? Do we know?
Russ, your flows are on surface. They are
not sure, Jody, but these flakes, these are halimeda flakes. So this is green algae. It's probably been disturbed by the ROV, velocity from the actual propellers, but it's actually mobilizing little flakes of algae, and that's what's drifting down through the water column in front of us.
We're moving you ahead. Yeah, Tony's down here on back deck, so it's kind of keep us first. Thanks, Bridge.
So just to let you know what's happening here, our umbilical floats have reached the surface of the ocean now. We're just above 100 meters down, so almost into the beginning of a recovery situation now. But we'll just keep looking at what we're looking at.
Yeah, thanks, Paul. We know we're getting to that. It's quite warm now. The red, I think, is probably these red blotches is the coralline algae, I would think.
Yeah, I think so, Rob. I agree with that. We're getting a lot of that now, despite quite a lot of sediment covering a lot of these ledges.
Yeah, but I bet a lot of that sediment is just halimeda. There's flakes that were they're just sort of drifting in the water column and they got disturbed. Yeah, you can see these. These are fronds, okay? So they're all joined together, like little corn flakes.
A couple of fish
the green,
there on the
of course,
screen.
is green algae.
Yeah, those look like fairy rasses to me. Stereolabris rosifascia.
Yeah, lots of halimeda here. Right, this is probably the dominant calcium carbonate producer just along these walls. The rest is just shallow coral that's been mobilized off the reef higher up. So you can see it hangs like curtains, and this is something we saw.
Fish on the right-hand side of the screen now. There's 3 different types.
I think we're also getting some hard corals, some scleractinian corals, some of the brown encrusting flat forms, maybe Parietes or Montipora or some of the agaricids that we've seen in some of the previous dives at these depths.
Yeah, that's right. And we have too, Leptoceras is another one. It's sort of disc-like, looks like a small plate, and brown. They've got this definite brown colour. But the thing is, the wall is so steep here compared to some of the other dives we've done. It would be a challenging place for a zooxanthellate coral to live.
Yeah, you're right, Rob. If we contrast this with one of the previous dives, I believe at Diamond Islets, where we had a much more gentle slope and substrate, and at this same depth, we had this absolutely extraordinary habitat of deeper water corals, algae, halimeda. So there's a real contrast with the
multiple fish on the screen right now, Kai.
Yep, cheers, thank you. I'm actually very fascinated by just how much halimeda deadfall there is. It just seemed to blanket the entire reef wall with white, and I'm just kind of admiring this quietly. Yeah, that looks like another chromis. I think Brian identified that as chromis degrayi previously. Hey, Jody, I have a question for you about Halimeda. It might be a silly question, but I know very little about marine algae. When they grow, are the tips characterized by younger growth, or do they grow from bottom up?
I'm not sure, actually. No, that's a question for Marty McNeil, who's working extensively on Halimeda. I'm not sure. Rob, any thoughts?
Yeah, they grow from the tips, so you can actually see them. They're older. You can see them, larger at the base, and they kind of bud off in places. I'm just trying to work out the Scleractinian corals here. I think these are these sort of browny plates that were starting to appear. It's plastered against the wall. There's one on the left that's called.
Rob, I think we just lost your audio. I think you were midway through a sentence.
Yeah, I was just saying that the Halimeda, you can see it potting off from the ends, from the tips. I just had a message from Tom Bridge. He'd love to join us, but he can't at the moment, which is unfortunate. This is definitely his zone. But look at the curtains of Halimeda here. It's almost completely blanketed at the background. That's all of these. Well, if it was on a flat area, you'd call it a meadow. I guess here it's just like a curtain, but it's all hanging down. And when it dies, it just creates this carbonate sediment that we've seen for quite a bit of this.
So the water temperature at the ROV is 25.8 centigrade, and when we last looked at the surface temperature on our sea surface logging, it's 25.7, so we're basically at sea surface temperature now.
Yeah, so we're definitely getting Scleractinian corals here. These zooxanthellate corals. These are these sort of brownish, circular plate-like corals. So these are true hard corals.
Okay, so we've just seen a cloud of these purple fish going past, but unfortunately I have to tell you that we're going to have to start recovery in the next couple of minutes. We're having difficulties with currents and, of course, heat. So we'll have to start bringing the vehicle in. I think we're going to take it out, dive it into deeper water, and then recover from there. Is that correct, Russ? Yeah. So that's what's going to happen. But we'll keep the feed on just in case we see something interesting.
Yeah, thanks very much, Paul.
Still this incredibly steep outcrops and substrates. It's a bit lumpier here, not quite as smooth.
The bright purple fishes just now were Pseudanthes pleurotinia, square spot Anthias.
Okay, that's it, guys. We're turning around and heading into deeper water prior to recovery. So that's it for the while, unless we see something interesting in the water column. But we're moving away from the reef now.
All right. Well, thank you very much, Paul, and certainly to the ROV team. We've really appreciated all your work today. It's been very interesting. Another interesting dive. Been very steep. For me, the highlight was probably seeing the different geological layers at the base of this big scarp, and certainly lots of interesting marine life in the nooks and crannies. Kai, do you want to wrap up? Any highlights for you from fish perspective?
Thanks for having me again. A great dive as usual. A lot of cool things that we saw. I'll send, was it Paul or Debs, timestamps for the interesting things that I want to get photos of. Yeah, but good dive. Good to have Jody here as well, joining us in the last leg.
Yeah, it was a real pleasure, guys. So thank you so much. I enjoyed that last part of the vertical face and seeing the really well-developed Mesophotic communities. I'm going to go back and review the base dive section with that stratigraphy and check that out myself later today. But have a great day, everybody.
Yeah, thanks very much, Jody. And just to talk through the plan for the remaining time here on this expedition, tomorrow we have another dive planned. That will be 367. We expect to be working on somewhere on the northern side of Lihou Reef. Again, so everything's weather dependent. It's still very blowy, but with Lihou Reef providing a bit of shelter, we should be able to do a dive in the morning before the wind really picks up. So it might be a little bit shortened. Following that, so that would take us to Tuesday. We'll have dive 368. That will be for World Oceans Day, and we hope to have a live stream with a larger group of people joining in from around the world and doing a live speaking while the ROV is in the water. So that's World Oceans Day. That will be the last ROV dive for this expedition. Following that, the ship will depart the Coral Sea Marine Park and head back to Cairns for a well-earned rest. So we're very much at the sharp end of the expedition with 2 more dives planned, and that will be it. So I'll sign out. This is Robin Beeman saying goodbye from Cairns, Australia, and I'm now handing it back to the Falkor's team. Thanks very much, guys, and goodbye.
Goodbye, Robin. Thanks very much for your excellent commentary today. And also thank you for Kai to join in and do the fish identification. And also Jody. I think Danny's still around, possibly. I don't know. But the narration is what helps make the dive better for everyone ashore. Thank you.